All right, thank you very much. The Vice Chancellor, Egerton University, distinguished ladies and gentlemen, I want to welcome you all to this two-day workshop that is organized on building institutional capacities on genetic engineering and biotechnology research in African universities and research institutions. It is organized by Egerton University in collaboration with Michigan State University in the USA, and we want to acknowledge the presence of Professor Song, who is one of the collaborators in this project. And this workshop, which we're going to have for 2 days, today, 16th and the 17th, is attracting participants from across the globe, and particularly from African countries. And we want to invite, therefore, all those who are here in person and all those who are joining us online. I want to recognize, for the sake of our presenters, those who are here so that we get to understand the people who are attending. We have representatives, the on person, those who are here in person, are mainly drawn from Egerton University, from several faculties. So I want to recognize those that are from the Faculty of Science. I see we have several. If you could just stand up, we will recognize you from the Faculty of Science. Thank you very much. We also have colleagues, that includes Professor Omolo, who is one of the key organizers of this workshop. We also have colleagues from the Faculty of Agriculture. Thank you. Yes, a good representation right there from the Faculty of Agriculture. We also have colleagues from the Faculty of Arts and Social Sciences. I know yes, thank you. So thank you very much. So those are the ones that are here with us in person. And we also have participants drawn from over 20 countries who had registered to participate in this workshop. And I see a good number have already logged in, and I believe they'll continue to log in as we continue with this meeting. So my work really is very brief. We're just going to recognize the participants, to welcome you all to this workshop, and say that as we look at building capacities, really we are looking to see how we can build capacities in terms of infrastructure, in terms of human resource, also the ecosystem for doing genetic engineering research, and also the social aspects. So, you know, when I look at the social aspects, issues of regulation and ethics, so that we have robust regulatory frameworks to ensure responsible genetic engineering and biotechnology. And for social aspects, just looking at public awareness and enhancing this, and the benefits and risks associated with genetic engineering. Many times when we talk about genetic engineering, what comes to mind mostly is the genetically modified foods and the debate that goes with that. But we are glad that we are having this meeting, that we can have open discussions to discuss what are the benefits and what are the risks, and how do we prepare ourselves? How do we foster global collaboration to ensure that we can harness the benefits that we can get from genetic engineering? So I want to take this time, therefore, to welcome the chair of this first session, who is the Deputy Vice Chancellor in charge of academic affairs at Egerton University. Welcome, Professor Aduda.
Vice Chancellor, distinguished participants, good morning. My role is easy. I would be inviting the speakers and timing them. But something reminds me when I see this topic, engineering, biotechnology engineering, or genetic engineering. Each year when the results for Form 4 leavers come out and the best performing students are asked, what do you want to do? Many talk of high-sounding words, neurosurgeon or engineering. But how come that I've never heard them talk about genetic what? Engineering, that they want to be genetic what? Engineers. They want to be biotechnologists. I think something is missing. I would hope that as we build the capacity, we not only build capacity at the top, but we also build capacity from beneath. So the extensionists that are in this room, make sure that after this workshop, we visit schools and other institutions and tell them that engineering is not just building cars and connecting what? Wires. Engineering involves a lot. In the interest of time, I would want to go straight to invite our Vice Chancellor, Professor Isaac Kibwage, to give his address. Thank you.
Thank you, Professor Aduda. Distinguished ladies and gentlemen, both physical here and virtual, receive warm greetings individually from Egerton University. As we gather here today for what I call commencement of the activities of the International Center for Genetic Engineering and Biotechnology, Regional Research Center, which is going to be housed at Egerton. It has taken a number of months, I think more than a year, since the proposal to host the center was done and the successful bidding to host it. Then we have gone through the commitment as the university and our government to put resources together that will support the operationalization of the center. I know that after we did the proposal and submitted, and the team from the International Center came and looked at our facilities, and we were notified that we shall host it. We were also given conditions that we needed to literally raise 25% of the value, because the way it was that the value of this center was going to be 5 billion plus. So as Egerton and the Kenyan government were supposed to bring in resources worth about 1.3 billion for the 5 years. So after many meetings and correspondence, eventually our government agreed. Remember, we are a government institution, so our resources are a part of the government resources, but on top of that, they agreed to give us 500 million Kenyan shillings for the next 5 years, obtainable at 100 million per year. So I would like first and foremost then to acknowledge the support of the Kenyan government towards what we are assembled to start off here today. I also extend gratitude to all of you for your participation in this event, which bears immense significance for the future of scientific research and development on the African continent. The theme of this workshop underscores the vital importance of harnessing the potential of genetic engineering and biotechnology to address the challenges and opportunities that lie before us. I don't know at what extent we should move on genetic engineering. Is it only for plants? Or we can also extend? Because when I looked as the director research was introducing, I didn't hear medicine. You know very well the fiascos, can I call it fiascos, in genetic engineering in what? In medicine. Diseases, new diseases, which actually would not be there if there was no what? Tinkering with genetics of viruses, of bacteria, and these are going to be with us for some time. So I think that as we set up, we need to also encourage researchers in genetic engineering of other materials than plants to be able to join us. Remember, we have been struggling with genetic engineering for HIV/AIDS for many years to create a vaccine, and we have failed. But we had COVID, and they came up with a vaccine. And both of them are caused by what? A virus. So what is so special about it that a vaccine can't be found, but we can find one for COVID? You know, these are the kind of things we'll be facing. And of course, when we talk about genetic engineering in the foods, what comes to the head first is GMOs. And you will be hopefully part of the participants that will remove or demystify that use of GMO food will actually alter the genetics of humans, because that's one of the biggest concerns, and therefore expose humans to problems of renewed diseases. Let me take a moment anyway to recognize and express my sincere appreciation to our distinguished guests and speakers, whose invaluable contribution will undoubtedly enrich our discussions and pave the way for groundbreaking insights. I especially acknowledge Dr. Banks, the director of ICGEB. I think he's online. Dr. Banks, are you online?
I am. Good morning.
You're welcome. Professor Yawa, our Director of NACOSTI. Dr. Yawa, are you okay?
Yes.
Professor Yawa? Okay.
Yes, you can hear me. You can hear me. Thank you.
Yeah, thank you. I was about to declare that you are not okay. Welcome. You know, not okay here means that you are not connected. Eh?
Yes.
Yes. Ladies and gentlemen, these two gentlemen have really worked tirelessly in giving me ideas and the consultations that have pushed us to this point. There is Dr. Ramesh. Ramesh, are you with us?
Yes, I'm here. Lawrence and I are joining together from New Delhi.
You are with Lawrence in New Delhi, eh?
Yes.
Okay, thank you very much for
thank you. Happy to be joining in this meeting.
Thank you very much. And I think let us be very thankful that because of the effort of Professor Song and Professor Molo, we are able to assemble here today. So, please, welcome. I know there are other distinguished guests, some will be joining us, some are online. So if I have not mentioned you, it is not because you are less important, but we have speakers, including Yasdan of ICGB, India also, Bernard Kolohoma of IAVI Nairobi, Oro Kenya, Dr. Mugira, CEO of National Biosafety Authority. I think him is going to be joining us this morning. Professor Patrick Okori, the Executive Secretary of RUFORUM. Professor Xiu Wang of Now China. I don't know what Now China means, but it must be somewhere in some authority in China. That's Nigerian Agricultural University. All right. My apologies. So, Professor Wang, are you on? Maybe it'll be on when it is his time to speak. We'll also have a chance to listen to Dr. Guchu, the Principal Innovations Officer at IUCA, and our own Professor Jojo Wall, the Director of SESAM. I don't see a wall here. Now, I acknowledge for your willingness to spare time and share with us your experiences as we map the way forward for this Center for Biotechnology and Engineering. The significance of this workshop transcends geographical boundaries, as evidenced by the many and diverse representations from African nations, extending even to countries in Asia. I think we have talked about India, the USA, and Iran. The enthusiastic registration of over 300 participants from 20 African countries shows the thirst for knowledge and the progress that unites us all as we battle the problem of food security, as we look forward to deal with the issues of hunger, issues of health, etc. I think we stand on the threshold of a transformative moment, both for Egerton, for Kenya, and for the African region. Therefore, the establishment of this International Center for Genetic Engineering and Biotechnology Regional Center at Egerton represents a beacon of hope for aspiring scientists. You don't have to try and get access to these facilities far away from home, but you will be able to travel within the continent, come to Egerton. It's a nice place, quiet. I think I like the serenity of it. And you can do research, because most of the time you do research, you know, it is cold, so you have no much running around to do. It reminds me of when I was studying in winter. It is cold, so you don't have this excitement of going around outside. So you stay more and do more. I think we already have research labs in this area within Egerton, which are running on. But we hope that in the coming years, we are going to obtain state-of-the-art equipment that will be able to get us started. We'll be attracting researchers to come with their resources, do research, and share the experiences and the findings with the scientific community. So through this initiative, therefore, we are not just fostering scientific advancement, we are cultivating for a future where a younger generation of researchers will have unprecedented opportunities to contribute to Africa's growth and prosperity. I urge each of you to remain focused and committed as we strive to achieve the objectives set for this workshop, particularly concerning the regional center. In conclusion, let us embark on this journey with a collective goal, unwavering enthusiasm and optimism. The collaborative efforts that will emanate from this gathering hold the potential to reshape the landscape of genetic engineering and biotechnology research in Africa. The ideas exchanged, experiences shared, and the networks established during this workshop will pave the way for a higher and more innovative and collaborative future. Thank you, ladies and gentlemen. Let us move forward together in pursuing knowledge and progress. Good morning again.
Thank you, Vice Chancellor, Professor Isaac Kibwage, for first of all welcoming us all to Egerton, and secondly, inspiring us and challenging us, particularly the genetic engineers and biotechnologies researchers, that we come together to solve human problems or humanity problems, particularly in Africa. We move on to the next speaker. As per the program, is Professor Walter Oyawa. Professor Oyawa has a long CV here. I'll just pick a few lines. Professor Walter Oyawa is the current Director General of the National Commission for Science, Technology, and Innovation, NACOSTI. He is a distinguished civil engineer with a PhD in civil engineering and an executive MBA. He has held senior management positions in Kenya, few of them being Deputy Principal, Multimedia University College, Deputy Commission Secretary at the Commission for University Education, Q. He has done research which is impactful, and he is an appointed ICGEB governor for Kenya and the local focal point for the establishment of the same. So help me welcome Professor Walter Oyawa to give his remarks.
Thank you very much, Prof. Am I audible?
You are.
Yes, the Vice Chancellor, Professor Isaac Kibwage, Dr. Lawrence Banks, Professor Ramesh, all the way from India, distinguished participants, good morning. It is quite a pleasure and an honor to be part of this great congregation. I think I'll be fairly brief. The VC haven't said much about this apartment center. For me, or for us at NACOSI, it's the culmination of a long process which commenced with NACOSI putting out a call for proposals where 7 universities applied, 3 were shortlisted, and Egerton University was unanimously awarded to host this very important center. I cannot overemphasize the importance of this center for the region, as the word indicates it's a regional center. We hope it will be really a regional center in Africa, and the second one after the one in China. I just want to take this opportunity to really thank Dr. Lawrence, who has really pushed for this, and also to thank the VC for the efforts, recent efforts that resulted in our cabinet secretary coming on board to support this center, pushing for National Treasury to fund this center. That is not easy, taking out of the current situation which our country is going through in terms of financial resources, and therefore it's a strong recognition that this center has a key role to play moving forward. As I speak, I'm attending the second working group meeting of the Biological Convention here in Geneva, and one of the discussions was the dual use of science and technology. And So think broader, think how you connect to such a program in terms of short courses, in terms of enlightening our people on these emerging things. They have the good side, but also the other side, if not checked, it might end up causing more harm than the good that we expect. But otherwise, I think I'm excited that it's culminated into this. I'm seeing a very distinguished panel, right from IUCA, right from China, from India, and even probably what Dr. Lawrence will mention, that Dr. Lawrence is in India to lobby for this center. Yeah, to lobby for this center. Yeah, there are a lot of minds on this center, and I'm very pleased that the Egerton VC is fully on board. Professor Molo, who has done a very good job as the lead scientist of this center, there are a lot of expectations. So we're also discussing you in Geneva here. Please do not let us down. So with those remarks, allow me to just say thank you very much and hand over back to the MC. Thank you.
Thank you, Professor Yawa, and we want to thank you for the support you have given to us as a university and to us as a country in ensuring that this regional center is located here at Egerton. The next speaker is Dr. Lawrence Banks, Director General, ICGEB, Italy. I'll read again his rich background, so I will skip some aspects. Dr. Lawrence Banks is the Director General of the International Center for Genetic Engineering and Biotechnology in Trieste, Italy. I think a place I've been a number of times. With an extensive career in, the eyes are failing me. Okay, in tumor virology, Dr. Banks is renowned for his groundbreaking work on human, now a physicist to read these tough words, human papilloma viruses, HPVs, and their roles in human malignancy. His expertise centers around understanding molecular mechanism behind HPV-induced cancers, particularly the activities of viral on proteins E6 and E7. Dr. Banks' research also delves into HPV entry into target cells, exploring the intricate interactions between viral capsid proteins and cellular machinery. His work has made contributions to many areas and also is involved in contributions to editorial roles in prestigious journals, reflecting his significant influence in the field of virology. Help me welcome Dr. Lawrence Banks.
Thank you very much. Can you hear me okay? Yes. So, Vice Chancellor Kawaki, Professor Ayala, distinguished guests, colleagues and friends, good morning and greetings from ICGB New Delhi. It is a great honour for me to be part of the opening of this meeting today, which I think is really coming at a critical point. Building capacity in genetic engineering and biotechnology for the benefit of the African continent. It's also very timely that this has been held at Egerton University, which I've had the real great pleasure to be able to visit. I really do agree with the VC's comment. It really is an amazing and beautiful place and a wonderful place to work. And it's timely because we're also celebrating the fact that Egerton will be hosting the ICGB Regional Research Centre for East Africa. All of us within ICGB are incredibly excited by this venture, and we are deeply grateful to the Costi and the government of Kenya, to Egerton University, and also the whole scientific community of Kenya and the region of Africa for their enthusiasm and support for establishing the Regional Research Centre at Egerton. As you all know, regional research centres are intended to act as hubs to promote top-class research and training, and serving the needs not only for the country in which they are hosted, but also for the region. In this Regional Research Centre, the focus will be on agriculture and biotechnology, and it will address the pressing need for food security in Africa. This can be achieved through a variety of means, involving traditional plant breeding and propagation approaches, in which Egerton excels, but also state-of-the-art modern molecular biology techniques, such as genomics, marker-assisted breeding, genome editing, to aim to improve both productivity, nutrition, and resistance to stress. And of course, the modern advances in the development of environmentally friendly biofertilizers and biopesticides. A major deliverable of the centre is getting these technologies directly to, and the improved varieties directly to the farmers and the people of East Africa. And we are excited by Egerton's capacity in being able to do this. Another fundamental feature of the RRC is to act as a focus for training and capacity enhancement. And we fully expect vibrant international exchange and research cooperation and fellowship exchange across the continent and beyond, as well as full participation of the Centre in the ICGB meetings and courses programs to directly offer training and exchange ideas and best practices in state-of-the-art agricultural biotechnology. And it was also mentioned at the very beginning the importance of reaching out to the next generation. And ICGB has always been incredibly impressed by how Kenya reaches out to school children and young budding scientists. And we really hope that the RRC will also be able to take part in these activities with ICGB in outreach to schools and local communities. With this, I thank you again for this opportunity and for the invitation to be here today. I very much look forward to seeing you all at Egerton and at Costi at the earliest of opportunities. And of course, I really look forward very much to seeing the Regional Research Centre operational and doing great science within the coming months. So thank you again. I wish you an enjoyable meeting.
Thank you, Dr. Banks. He has stressed the great responsibility we will have as an institution in ensuring that the outcomes from this center are propagated to the people who need it most, farmers, etc. So thank you. We move now on to the keynote speakers. And the first keynote speaker is Professor Ramesh V. Sonti, Director, ICGEB, New Delhi, India. I will read again a brief of Professor Ramesh Sonti. Dr. Ramesh Sonti, Director of ICGEB, New Delhi, India. Since 2023 January, is a distinguished scientist with a rich academic journey. His degrees are from prestigious institutions, and he has a PhD in bacterial genetics from the University of Utah, USA. And he has excelled both in research and leadership roles. He has also received awards like the Shanti Swarup Bhatnagar Prize. Dr. Sonti's contribution also extends to bacterial disease-resistant rice varieties, earning his team esteemed accolades like the CSIR Award for S&T innovations for rural development and the Biotech Product Process Development Commercialization Award. His expertise brings invaluable insight to the ICGB and the workshop discussions. His topic is importance of capacity building in genetic engineering and biotechnology. I hope he will do it from where he is. Thank you. Let us welcome Professor Sonti.
Thank you, Professor Aduba, and my thanks to Professor Isaac Kibaje, Vice Chancellor of Egerton University, and Professor Walter Oyaba, who preceded me, and our Director General, Dr. Lawrence Max. It's a pleasure to be joining all of you today. I would like to share a short presentation on the importance of capacity building in biotechnology and genetic engineering, what it would mean, what I think, what ICGB has meant for India, and what I feel ICGB can actually mean for Kenya. So if I can actually share the presentation, please. Can you see the presentation? Yes. Okay. So I will give a very short presentation. And at the end, if there are some questions and answers, I'll be happy to answer. Next slide, please. So what I've tried to address over the next few minutes is on why Kenya and East Africa should build capacity in biotechnology. I'll sort of try and address a few important points about why it should be locally grown rather than having technologies imported, developed elsewhere and brought into India. And I'll very briefly touch on what are the kinds of biotechnological solutions that we have developed or are developing at ICGB New Delhi, and that have implications and have had impacts within India, and hopefully in the years to come, the RRC actually have a similar kind of impact for East Africa. Next slide, please. See, there are three major reasons I feel that there should actually be RRCs, an RRC should be set up in Kenya. Why capacity should be built up in biotechnology and genetic engineering. And the foremost is that there should be local solutions for local problems. And I will elaborate on that as we go by. The second point, very important point, is on optimal utilization of national biodiversity. Biodiversity is a treasure and a wealth of each country, and how best the biodiversity skill that is present in any country will actually benefit the people of that particular country. So I feel that as it is a treasure, the utilization of the treasure should actually primarily come from the nation in which that treasure exists. And finally, very briefly, I'll talk about circular bioeconomy for Kenya and why genetic engineering and biotechnology capabilities needs to be built up in Kenya to enable, in the years to come, the development of a circular bioeconomy in Kenya. Next slide, please. Now, I just want to give you one example of how we envisage ICGB can help in the development of local capacities and technological solutions in various African countries. And just I'll give you an example of how we're working with a research organization in Nigeria on enhancing shelf life of tomatoes. So they had expressed an interest. There is an interest in Nigeria for enhancing shelf life of locally grown tomatoes using the technique of genome editing. Now, I will not go into the details of genome editing or transgenic technologies because Professor Song is going to be speaking after me on each of these new technologies. But there is this capability, technological capability, that has been developed recently along the last 10 years or so to be able to go into the genome, the DNA of an organism, and directly change in the genome certain constituents of that genome, basically the base pairs that are there in the DNA, you change them directly in the genome to impart environmental characteristics for crops. The same can be done for humans to correct certain inherited diseases, but I'm primarily focusing on the agricultural aspects. So this technology is being actively used in New Delhi for improving various crops that are important in India. And there is one particular laboratory of my colleague, Dr. Snehalata Pare, who has established these genome editing technologies. So my colleague was interested in collaborating with the researchers in Nigeria to enhance the shelf life of the tomatoes, the tomato cultivars that are being grown in Nigeria. The difficulty is that the biodiversity authority rules in different countries have certain regulations on transfer of varieties from one country to another. So upon discussion, what we felt was that the best thing would be if researchers from Nigeria would come to New Delhi, get trained in New Delhi in these technologies, working on tomato, but working on varieties that we are working on here in New Delhi, learn the technologies, and then go back to Nigeria and work on the varieties that are actually being grown in Nigeria. So the variety doesn't have to be brought from Nigeria to New Delhi. It's just that the capacity, the researcher gets trained here in New Delhi and is now capable of actually doing these, empowered to implement these technologies in Nigeria. So we are actually having a researcher very shortly coming from Nigeria to ICGB New Delhi on an ICGB fellowship to gain experience in genome editing of it. So we hope that once the researcher learns this technology, upon return to Nigeria, now the researcher will be performing genome editing on local cultivars of tomato. So the infrastructure is already available in Nigeria. All that is required is that we provide the capacity building from New Delhi to our Nigerian collaborators and friends, and then they go back and do the research there. So what we envisage is that in the years to come, as the RRC in Kenya develops, we will have these exchanges of researchers from Kenya to New Delhi, possibly to Trieste or in Cape Town, where we learn some of the newer technologies that have already been implemented in ICGB cooperation countries, and then go back to Kenya and actually implement them. So this way, we don't have to exchange the genetic material, because the genetic material is part of the wealth of each country. So the genetic material doesn't have to be exchanged. We just build capacity, and then the activity can be done in their own country, in this case, India. So this is one example that we hope will be very fruitful and will benefit our colleagues in Nigeria as we move forward. So this is one example where we feel is a successful way in which you can see how capacities can be built in Kenya as we go along. Next slide. Now this is another example that we've recently been interacting with a colleague in Ethiopia. Sesame is an important oilseed crop of Ethiopia, and there is a serious bacterial disease of sesame in Ethiopia. This disease is called bacterial blight disease. So the colleague in Ethiopia was interested in coming to UAE because we have expertise in working on bacterial blight of rice. And in this time, I can tell you a little bit about the work that we have done in this area, where we have developed a disease-resistant variety, a rice variety that is resistant to bacterial blight. The problem for us has been that bacterial blight is not a serious disease of sesame in India. And we have inquired with various research organizations within India, and it turns out that nobody in India has a strain that causes this disease on sesame. Because the disease is not a problem, there has been no efforts to actually gather that strain, and we were not able to get that strain. Now, importing, just go back to please. Yeah, importing the pathogen into India is an elaborate process, especially because the disease doesn't appear to be very serious in India. So bringing in a new pathogen into India requires, there are procedures in which it can be done, but it's a very elaborate process. So it would be best if the problem is tackled in Ethiopia with training of researchers in the appropriate technologies at ICGB. We're running, since it's in the context of the ICGB RRC, we're thinking about ICGB, but it could be anywhere else, for example, Michigan State University or any other organization. So I think the best way is that because of these kinds of issues, that there are varieties that are adapted to local conditions, and there are restrictions on movement of germplasm between national boundaries, and also because there are certain diseases that are local, that may not be a problem in other geographies. Exchanging the pathogen may be difficult, so it would be best if local solutions are found to local problems. And that is one big reason why there should be an RRC in Kenya to take care of problems of Kenya and the East African region. So this is one area. The next slide, please. Another area is about the utilization of national biodiversity. As I was mentioning, biodiversity is the wealth of each golden niche. And it must ultimately be used to the benefit of the people of that country before it is moved on and hopefully used elsewhere in the world. Now, one aspect of biodiversity is that biodiversity, the richness of biodiversity lies in the fact that it is locally varying. So, for example, a colleague of mine, Dr. Vittorio Venturi, works on soil microbiomes. And he works in Trieste ICGB. And the soil microbiome, the microbiome is a combination of all the microorganisms that are present in the soil. It's not just one species of microorganism or two species of microorganism, but it is the entire hundreds of species, many more, of microorganisms that are present in the soil. It is a very, very important component of soil health, and the vibrancy of the health of the soil can be maintained only if the soil microbe is also conserved. So, Many researchers all over the world are trying to isolate microbes from soil and then add them to plants to enhance their growth, their yield, and their ability to resist abiotic and biotic stresses. The difficulty again here is that the soil microbiome varies locally and regionally. So a microbe isolated in New Delhi is unlikely to work in Kenya. And a microbe isolated in Kenya is unlikely to work even if it is the same crop, it is unlikely to work in real estate. So it is important to understand local soil microbiomes to effectively use them for enhancing plant growth and protecting crops against various biotic and abiotic stresses. And we hope that down the line, the RFC in Kenya will take a very important role in understanding soil microbiomes in Kenya and East African countries, and ultimately use this particular microbiome, the microbes that are isolated from the soils in Kenya to improve the crops that are being grown in Kenya. Next slide. The other aspect, and this is something that we were also thinking about with regard to the sesame project that I was mentioning in Ethiopia, because there are varieties of sesame that are tolerant to this particular disease in Ethiopia. So those varieties are going to be very, very useful, not just for Ethiopia, but everywhere in the world. Similarly, there may be plant extractions from Kenya that have important traits that can be exploited to develop newer varieties of crops elsewhere in the world. So the utilization, the capacity to utilize the biodiversity of the country should exist within that country. And the RRC in Kenya, we hope that down the line, it will essentially be able to utilize the extensions of important crops of Kenya that are present within Kenya, identify the traits that are important, and so that they can be exploited to develop new varieties of crops. Now again here the important point is not only the wealth that is, the genetic wealth that is present in Kenya, it is also important that the varieties of crops that are grown in Kenya are going to be different from the same crop being cultivated in South Africa or in Nigeria or in India. So the local varieties have to be improved, and the traits that are present in the crops that are present in Kenya should actually be exploited. And any benefits that come must first come, any benefit that comes from the utilization of genetic resources of plants should first come to Kenya. And then it can be used elsewhere also. So it should not be that the resources of Kenya are used for benefit of some other country and not, so the local capacity to understand how much variability there is, how to utilize that variability should be developed within Kenya so that it can be used for crop improvement in Kenya as well as elsewhere. But the other aspect is that there is a lot of traditional knowledge about traditional medicines in Kenya. And this knowledge is coupled with plants that are present in Kenya that can be medicinally important. So all of that knowledge must be leveraged. This RRC in Kenya should actually work towards harnessing this knowledge, utilizing these resources, and hopefully develop newer varieties of crops and newer medicines for the benefit of Kenya and elsewhere. Next slide. Now, the last slide is about, the last really important point I want to bring in is about the circular bioeconomy. All over the world, there is a movement towards the bioeconomy. Now, what is a bioeconomy? The bioeconomy is an economy in which every activity within the economy is made through biological resources. So if it is an enzyme, it is a food, it is a medicine, even a pharmaceutical compound or a fuel that is made should actually be derived from biological material. The raw material should be biological, the end products are derived from biological activities, activities of biological resources. The reason is that anything that is derived through biology essentially will be degradable. It is a biodegradable component because it is already been seen in nature. There will be organisms that will be able to convert that final compound into its basic raw materials and put the resources back into the economy. So it is a circular economy where resources are utilized effectively and in an environmentally friendly manner. And in the years to come, this is the future of the world. In countries all over the world are moving in this direction, including in India, there is a big push towards a circular bioeconomy. And in the years to come, this will actually be an important activity. If it is not already an important activity in India and the East African region. And what we are finding in India is that any biotechnological solutions that you develop will be, for the circular bioeconomy, will be developed using local bioresources. So any solutions you make must be tailored to those locally available bioresources. So the circular bioeconomy in Kenya will require tailoring of any biotechnological solutions to be able to work with the local bioresources that are available in Kenya. So capacity building in biotechnology and genetic engineering is going to be a very, very important investment for Kenya. Next slide. Now, what has ICGB really has done over the last 20-25 years? Other than in capacity building, what are the products that we have actually developed for India? One of the points I want to mention is that the diagnostic kits for dengue that is used worldwide, about half of the kits made worldwide, utilized worldwide, are actually from technology that was developed in ICGB New Delhi and licensed to a company in India. That company sells that product in 45 different countries all over the world, including in countries in Africa. We've also developed with the same company kits for diagnosis of hepatitis C, HIV, and celiac diseases, and all of these are in the market. We've also developed a botanical drug and a vaccine for dengue that have been licensed to the industry. And the industry is actually taking forward for additional clinical trials. An anti-malaria vaccine that was initially developed in ICGB New Delhi against Plasmodium vivax is now entering phase 2 clinical trials in Thailand. These trials are being conducted by the Institut Pasteur in France in collaboration with Oxford University, because the faculty members who have developed the vaccine here in ICGB New Delhi have now moved on to join the Institut Pasteur in France and is continuing to develop it further with collaborations here. Now, very recently, in response to the SARS-CoV-2 pandemic, a colleague of mine in ICGB New Delhi, Dr. Anurag Chandran, what she has done is that she has developed monoclonal antibodies, therapeutic monoclonal antibodies that can serve for a therapeutic purpose against SARS-CoV-2. And the interesting part is that these monoclonal antibodies appear to be working against every variant of SARS-CoV-2 that has been identified so far. And this is something that we are actually testing it further before we license it out to a company. In ICGB New Delhi, we have developed genome-edited rice lines that are higher yield. We hope that next year they will go into national field trials within India. We have developed more stress-tolerant rice lines that are more tolerant to disease, more tolerant to abiotic stresses, and very importantly, we identified very recently, and these are things that we are working in partnership with national research institutes that work on rice. We have identified some lines that appear to have very high levels of iron in the endosperm. Most of the iron that is present in rice is present in what is called the alveolar layer, which is the upper coating of the seed. And upon milling, that layer gets removed, and the iron gets passed along the alveolar layer, and we essentially lose that material, or we feed it to our poultry or livestock. The goal for rice breeders within India is to make a biofortified rice that has high iron in the endosperm, so that which is the part that remains, which is a grain of rice, it still remains after the milling is done. And that has been something that I've been looking very hard within the Indian agricultural research system to find such rice lines. And my colleague Dr. Tanushree has recently used genome editing to enhance the levels of iron in the endosperm. So we are trying out these lines in collaboration with our partners in the agricultural research system in India. And if they are validated again, they will be going into field trials and hopefully will join in national food programs to eradicate iron deficiency within India. We also developed herbicide-tolerant transgenic rice lines. That will again be something that is important as we move forward. Very important activity that we have done is that we've developed certain enzyme cocktails for degrading agricultural agri-residues. Now, after the harvest is done, you collect the grain, but the bulk of the material that is present in the environment is sometimes hopefully used as feed for livestock. In other cases, it is actually burnt. And in India, that is a serious problem in the northwestern part of India, because after the rice crop is completed and the next, before the wheat crop is, the subsequent wheat crop is planted, the rice fields are being burned, and that leads to pollution within northern India in neighboring countries in the winter months. So we developed certain enzyme cocktails that would help in degradation of the cellulose material that is present in this agri-residue and to convert it into biofuel. And we are trying to work with the industries within India to be able to see if this can be commercialized, so that we will contribute to the circular bioeconomy that I've been mentioning. Next slide. So what kind of infrastructure is needed for RRC? Now what I'm actually putting in is what I would say is the minimum that will be required. You need infrastructure and capacity for, biotechnology requires the use of enzymes, and these enzymes are very sensitive to temperature, and they work in specific conditions, specific temperatures. So there have to be laboratories that are air conditioned, and there should be backup power. The backup power is required because if the air conditioning fails, all these experiments and reagents that are very expensive, if the power goes and there are temperature fluctuations, all the amount that has been funded, that has been all the money that has been spent in buying these resources, and the tremendous effort of researchers over several months of work will essentially go bad if the power goes. So air-conditioned laboratories with backup power should be available, and equipment for routine molecular biology, biochemistry, and microbiology. Now, it would be ideal if there are facilities, advanced facilities for genomics, proteomics, etc. In the first stage, even if they are not available, these can be outsourced. In New Delhi, we outsource for genomics studies to companies that provide these services. So while it will be desirable to have them, they can also be outsourced. What is very important is to have, since Egerton is going to be focusing initially at least on crop biotechnology, there should be greenhouses for growing the crops under contained conditions. There should be growth chambers that define specific environmental conditions, humidity, temperature, etc., and the day-night cycle in which crops are grown. There should be tissue-culture and field facilities. If the RRC would also be enriching studies on, as the Honorable Vice Chancellor is mentioning, if there would also be additional studies to be done on medicines, then animal house that will hold animal models for research will be required. Wilson 2 or Wilson 3 facilities, depending on the level of containment that is required for working on pathogenic infections organisms, that would also be required. Next slide.
Now, I was also saying that biotechnology is changing. So the capacity that is built must be such a way that there is scope for retooling constantly. So I would say that in my 30-year career or so, I've had to reinvent myself 3 times. So every 10 years I will reinvent myself because the technology has changed and now I have to learn on this. So not only should, so training and capacity building is not a one-time effort. It is a continuous and constant effort. Because the technologies themselves that we use in biotechnology, they are constantly seeing improvement. So the omics technologies, genomics, proteomics, metabolomics, all of these are constantly changing. So one needs to actually be able to develop these capabilities constantly. Now, new techniques of synthetic biology, where you now design cells and tailor them to have new kinds of functionalities, these are again coming. The synthetic biology techniques use omics and gene cloning and genome editing technologies. So these are again things that have to be constantly learned and improved upon as we move forward. The latest key to the block in terms of biotechnology is the application of artificial intelligence and machine learning tools along with omics technology to lead to many new applications. So there is a need to constantly redo. So this is something that we have to think about, and the capabilities, the individuals, the researchers who work in this area must be ready to actually, must be ready and eager to learn new technologies as they get developed. Next slide. Now, what do I see as the prospects for the RRC in Kenya based on my own experience or what ICGB has done in New Delhi? Building local capacity in biotechnology and genetic engineering so that these technologies that are relevant to the local economy are developed and they are absorbed and help the local. That's number one. And ICGB can help in the capacity building, the training, handholding, and these are not only researchers who come to New Delhi and we also go on site and conduct workshops and so it's not a one-time exercise. We are committed to actually helping you in this journey. And together, I think ICGB and the authorities in Kenya and RRC in Kenya, we can work to enhance the contribution of biotechnology and genetic engineering for the betterment of the economy of Kenya, East Africa, and all of the people who live in Kenya and East Africa. I think I'll stop there. Thank you. Thank you very much.
Okay. Thank you very much, Professor Ramesh Sonti, for the insightful lecture. And in your ending remarks, that we have to continually renew ourselves. I think that applies not only to the biotechnologists here or the genetic engineers, but all of us. I know time is spent. If there is any two burning questions, I allow now short questions to Professor Ramesh Sonti. Any? There is one.
I have a policy question.
I think that there is.
Can you hear me?
Yes.
Hey, Professor Sandy, glad to see you. I have a policy question. It's like, good, and you? Yeah. My the policy question. In India, are the gene-edited product and the traditional GM product, are they similarly regulated?
the regulations for commercialization of genome editing crops has been recently changed in India. What has actually, the new rules say that for genome editing you have to make the transgenic construct, but after the genome editing is done and the transgene is removed through segregation, the genome edited crop can be considered to be equivalent to traditionally bred varieties. So the rules for commercialization of genome editing crops have been substantially liberalized. And we hope that in the next year or two, these products will start hitting the market in India. Thank you.
Thank you very much.
One final question, if there is. Okay, I can't see. So we thank you, Professor Ramesh Sonti. We move for this session. The keynote speaker is Professor Guo Quinn Song, and I will read a brief of Professor Guo Quinn Song. I hope I'm getting it correct. Yes. Yes. Okay. Professor Guo Quinn Song is Associate Director of the Plant Biotechnology Resource and Outreach Center at Michigan State University. And he is a prolific researcher in plant biotechnology. And his research spans genomic improvements of horticultural crops, deciphering molecular aspects of flowering and engineering genetically modified crops for yield enhancement and abiotic species and the others. His presence at the ICGEB RRCC workshop promises valuable insights into advancing genetic engineering and biotechnology research. So,
okay. Hello, everyone. Can you hear me well for those online? Okay, great. Yeah, first of all, I would like to thank Professor Omolo and Egerton University for organizing this workshop. It's really a great pleasure to be here, and I'm so excited to see all of you. And today I'm going to talk about new biotechnology tools and strategies like for agriculture. Okay, so I'm a biotech person. I have been working in this area for over 20 years. And for this workshop, I think I would like to thank Michigan State University for sponsor this through this AEP program, and that allows me and Professor Omolo to work together for this institutional capacity development in the area of genetic engineering and biotechnology. So my focus today is going to give you open the window to see a little bit, to see why we need biotechnology. What are those biotechnology tools? So the first question is, well, for those of you who are not working in biotechnology, either for plant, animals, humans, is very, very broad, right? My focus, I have a background in plant sciences, so my talk is focused on plants. But similarly, the tools are the same for the other animals, human studies, all the others, okay? So in terms of plant biotechnology, this is a very broad scope. It's not only for genetic engineering. It can for germplasm conservation, micropropagation, you can name, okay? So I'm not going to go into the details, but keep this in mind. Biotechnology is a very, very powerful tool for genetics, genomics, and the other, like micropropagation. So the first question we usually ask ourselves, why do we need to alter plants? Why do we need to alter animals? Why do we need this? Because we know the population is growing, right? I know most of you or all of you knows we all get like the population like reach like 9 billion by 2050, right? This number might change, but that's the number one is population is growing. Number two, we have already felt that global warming, climate is changing, right? This is critical, right? We know that I'm not going to go to these numbers, but we already felt that. This year, like in the U.S., there's several days the temperature is just like break the records. Okay, global warming is with Earth. Okay, and then meanwhile, when this happens, the natural resources is decreasing. We get more population. We get the climate changes, and if you look around, we see the arable land is decreasing, right? So while use biotechnology as a tool, because this chart is showing, right, on the top is traditional, this is breeding, you make crosses, and then you pick up the cultivars or the lines we need, right? It really takes a long time, and meanwhile, when you do, okay, we have a trait, a drought tolerance. This trait, if we throw traditional breeding, it's usually several years, several generations. It's very, very painful process, especially if it goes to the woody plants. Genetic engineering is is pretty strict. Okay, I have a gene, this orange one. I know that. Then you take this gene out, put it specifically to this, this green one. This green are the desirable genes, we call that, okay? And then it's very easy to do, okay? So that's why biotechnology is desirable, it's powerful. That's why it represents something new for the future, okay? So, what's the current standards of the biotech crops in the world? So briefly, I put this information together, okay? Started from 1996. Right now, there are 4 major GM crops. It's like corn, soybean, right? Cotton, and canola. And actually, right now, in the world, about 50% of soybean, they are transgenic. Okay? Not going into the details. If you look at the data year by year, there's about 10% annual increase in the past over 2 decades, right? The major trait, if we see the GM plants, the major trait is herbicide resistance and sometimes insect resistance. And right now, there's a stack of the insect resistance and herbicide resistance, not many others. Okay, these are two major traits that's in the GM crops. And if you look at the map of the countries that have adopted GM crops, there are about 30 countries in the world. We know that in the European countries, most of them, they don't accept GM crops, right? In Africa, you can see Sudan. There's 2 countries over there. But of course, sorry about that. We don't have Kenya on this, right? If you see here, there are 2 countries over there. So about fruit crops, okay? This is this. We debate a lot, and there is controversial too. So right now on the market, transgenic papaya was the only commercialized, GM fruit crop, because there's no other option. Virus-resistant papaya. If we don't adopt that, we don't have any. Probably, we don't have enough papaya fruits, right? And then right now there's in the U.S. it's already commercialized like a non-browning apple. They use a genetic engineering approach. Okay, not many like the others are. I was asked many times by my friends because I work in this area. They ask me, "Is that big peach is GM peach?" No, the technology is not there. Even we want to get the GM peach, we don't have the technology developed. If we say, look, what are the new tools for the GM? Okay, this is from a Nature Biotechnology publication. It's summarized like, okay, gene editing. Gene editing is new. It's very exciting. It can be TALEN, CRISPR. But anyway, I will give you more information about gene editing. Then it's called a new term like intragenesis. Why is it called intragenesis? It's like I transfer apple gene to another cultivar. It's instead of like I transfer a gene from a bacterium to corn. Okay, it's called intragenesis. For horticulture plants, and grafting. There, because the horticulture plant is the rootstock and the sign, you can engineer the rootstock to change the sign. All this is help to minimize the concerns about the GM crops. Of course, there's no evidence shows GM crop is not safe. Okay, it's just concerns. Concerns is there, but we don't have scientific evidence to support GM crops. They are not safe. Okay, so we talk about biotech. Definitely, we are concerned about genetic engineering. Okay, genetic engineering actually is just like a short summary. Like in this slide, you take a gene, you do engineering. What means engineering? It's like we build a building. You put like a puzzle piece of the genes, put it together. You say, oh look, I want this gene to increase the yield. Then you make it. After that, the next step is I want to modify plant. Then I put it in plant. You can use agrobacterium. You can use gene gun to put it in, and then to modify that. Of course, this can be used for animals, right? So I mentioned like intragenesis. The GM crop, they call the GM crop because they are transgenesis. What means transgenesis? Because those herbicide-resistant gene or pest or insect-resistant gene, they are from bacteria. Okay, so to overcome this type of concerns, like we can isolate the gene from the plant, we can transform this to another genotype. Like I isolate one from rice, you put it in rice, then you get like cisgenesis or intragenesis. Okay, so like this is the difference. It's very, very clear here. Transgenics, traditional GM crops, you get a gene from bacterium, you put it in apple. This is transgenesis. You get a gene from corn, you put it in apple. This is transgenesis, transgenic. Gene editing is like, okay, I have a green apple. I don't put anything new. I just use my tools to change something. Okay, then you get like a gene editing product. Intragenic, as I said, you get a gene from one cultivar to the other one. Like I get a gene from a green apple. Eventually, I transfer to another cultivar. Then I get a red apple. This is the difference. Okay, I'm saying this is the new technologies like gene editing, intragenesis, transgrafting. All of these new techniques allow us to, for the future, help the product to be easily accepted by the consumers. It's easily to get this marketed. Okay, this is why new technology is desirable. Okay, gene editing, right? We all know this. It's true. It's there because the scientists they are awarded Nobel Prize. It's 2020. I don't need to explain too much about that. It's true or it's not, okay? It must be true, okay? And then gene editing, as I said, we don't need to put any extra DNA from the other species. You just go directly inside of your local cultivar or whatever. You want to change that, then we do it, okay? So this is gene editing. The regulator, I asked the Professor Sonia a question about regulatory question. This is regulatory, like this is, well, what we are doing is what we are doing now is not to convince the scientists. For most of us in this room, we know they are safe, but how we are going to convince this general public they are safe, they are safe. Okay, there's sometimes mistrust science or something. Okay, the regulatory thing, in fact, gene-edited plants, it can be GM if there's some parts. It really depends. Okay, in general, gene-edited plants, if you don't input any transgene, they are completely safe. It's similar like traditional breeding because we don't have a way to find out if it's changed or not. Okay, when we do traditional like crossing, we cross it. Do we know how many genes are exchanged? We don't know exactly, but we can find that out. Okay, so this is like some products is already is being tested in the field, like in the U.S., in England, in China, and Japan. Okay. Oh, my slide is frozen. Okay, it works right now. Okay, I'll give you some examples about like the GM application, GM technology application. It can be used like to improve like the quality of the plants. Okay, this is a golden rice. Some of you heard this along this story, right? And then it can be used for biofuel. It's a hot research area in the U.S., especially when Bush administration they started this because there's concerns about the fuel, like it's consumed very fast. Then they try to develop biofuel using genetic engineering. Okay, for the traditional, for agriculture, we really say like GM for disease resistance, pest resistance, abiotic tolerance. Okay, or for like some time to enhance or to boost like the yield potential. Okay, so biotech capacity development. This is the same of this workshop. Okay, so what type of capacity do we need? If you look at the sequential steps for plant genetic engineering, or as well as other genetic engineering, there are the 4 major steps. I usually use this slide for my teaching. I told my students, okay, in order to do that, you're going to have a gene. What are we going to engineering? That's the gene, the first step. The second is you use the gene cloning technique to get the gene, get the piece of the desirable trait. And then you do a genetic transformation. That's based on tissue culture or whatever. Okay? Then we do field test, greenhouse test, and field test, and then go to commercialization. Okay? This is, for example, like, okay, I cloned this gene. This is for cold tolerance, and this type of work is not like operated completely free. Even in the U.S., this is under-regulated. Okay, there's a different levels at university level and like U.S. federal level. These are the agencies regulated this type of research activities. So if you look at the facilities in the U.S., this is published in Plancer. Like in the U.S., there are six major facilities like I have here. We open the service to the other folks, to the other people. Like, okay, if you want to do some genetic engineering, you don't have the capacity, you don't have the techniques, we do that. We provide that service. Okay, I know that it's hard to read, but you don't need to read. You just know in the U.S. we have these resources. It's open, but not for all. Not all the universities have this. Okay, my center is like the highlighted one. Okay, so why I'm saying the biotechnology is very, very powerful. The tools, these are the tools. I'm showing these plans. That means these are the tools we have. We can do all this. My center is called Plant Biotechnology Resource and Outreach Center at Michigan State University. It started from the year of 2000. I joined in this lab in 2002. So far, we have engineered or transformed 27 plant species, including many of these are very, very important to Michigan. okay, like blueberries, cherries. Why we did cherries, blueberries? Because they are important to Michigan. We are not working on avocado, we are not working on coffee, we are not working on Kenya tea because they are not our local plants. So at the local level, we do need a facility development here, okay? So like this, we developed this, these are new tools. Probably right now, we have the best like tools for doing this, like root bird, and like sweet potato, all of this, dry bean, okay? So the capacity, if you have the facility, so there is like the capacity, it can be different levels, technical level, training level, education level. So we did all this. You see here, this is like a Bollock follow in my lab. I give you an example. This is a visiting scientist from China, and this is like from Iraq. She is like a graduate student, and then this is a postdoc and like undergraduate students. This is like the similar workshops I give like every year to the visitors over there. Okay, and then like this is a Nanjing Agricultural University. I went there 2019, right before COVID happened. Okay, these are a group of graduate students. They are all interested in biotech. Okay, that's why in China it's developing very fast because many young students they are pursuing this is kind of a fashion thing, like we talk about gene editing in biology. Okay, sorry. And this is like a gentleman from New Zealand. She just traveled a long way to my lab to learn blueberry transformation. Okay, it's the training process. We can do all this type of short course like workshop. The purpose is to help whoever needs this technology. So give you a little bit very quickly, like what we have done. Why I am thinking this is powerful. This can do a lot of things that cannot be done using traditional way. Transgrafting. We developed protocols for transform of cherries. Okay, I don't think we grow cherries here, right? Cherries is a favorite fruit for many people. It's very difficult for transformation. We developed that, but we know the consumer is going to concern about the product. So what we did, we engineered the rootstock, the bottom parts. So what we did, we put a virus-resistant gene in the rootstock. We use a strategy called RNA interference. That's another technology. Was awarded the Nobel Prize in 2006. Okay, so the scientific background, there's no question. So with that, the small RNAs produced from the rootstock, they can be transported to the sign, and then after it's transported, the virus starts to attack the sign. The sign part has the resistance because the small RNAs. Okay, this is one example. Similarly, we did blueberry. We transformed many of these. Each of these like is a transformation events. We do that for freezing tolerance, for herbicide resistance, for early flowering, for yield, and we are not doing gene editing. Okay, not going to go into the details. This is the way like, okay, we get early flowering blueberry. That's on the top is the gene. That's the target. You put it in blueberry, you see on the left, you see the tiny plants flowering. You can make them flowering while the non-transgenic, they can never flower without cheating. This gene is from blueberry. I just changed the expression of the gene. I put it in the other way. Well, these new materials can be used for fast breeding. Like, okay, in traditional breeding, blueberry, you need 3 to 5 years. But if you use my materials, you can do this 1 year because you can let the plant flowering 1 year. Okay, this is another application. This is freezing tolerance. Okay, I get a blueberry gene that's like and overexpressed in the other one, and eventually you get like a transgenic one. This one, you see the green leaves, these are transgenic. The wild type, they died after the freezing treatment. It's so obvious, okay? The technique is there, the product is there. We haven't started to commercialize this, we haven't found a way to do that yet. But I'm just showing you the technology is very powerful. It can be a lot of things we can do. For the yield, by doing genetic engineering, you have to have some information about genetics, genomics. Then you can identify a gene, a new thing. This is our K-domain technology. From our research, we found like a piece of DNA that can increase the yield. We call that yield enhancement instead of yield protection. Yield protection is just like, okay, herbicide resistance or like drought tolerance. That is just to protect the yield. But this one is enhance the yield. So this gene is from flowering pathway. My research area is mainly in flowering pathway. It's trying to increase the yield. We know the yield is important, right? We get more population. We need to secure food for our future, okay? So gene editing technology, this is a collaboration with the USDA, okay? They spend a lot of money. Right now, we are developing a new gene editing technology. It's almost there. You see the transformation system, it's very promising. We are hoping that by some day later, we can have the tool development, we can manipulate the grapes, change the color, change the flavor, change the tolerance, and improve the yield. So that's the main purpose. Okay, so that's some examples. Just briefly, let me back to the focus of this workshop. I know we are going to have a lot of good discussions, how to operate, how to get the work done. So this is like from today and in these 2 days, I'm pretty sure we are going to have a lot of interesting discussion. Okay, so and then we can figure out a way for the future, for the next steps, and. Again, like this picture, I took this yesterday. Okay, so I was thinking, why I come here? This is like to feel something real, to see something real, maybe to say, I am very glad to see Professor Saide in the audience. Okay, she knows MSU. This AEP program is not the first here. It's at least, to my knowledge, Dr. Wayne Spears was here. He's the director of the PBROC, and also he's the chairperson of our department. So we have a long time, it seems to me we have a long time of collaboration with Egerton University. So I hope, for the future, for next steps, we can find a lot of ways to collaborate, like for the research, for training. We can do a lot of things. At this point, I'm very excited about that, and I'm very, very happy to see all of you, and that's all that I have for today. Thank you very much.
Thank you. Some of us are learning new terms and new things every day. Yes, so in the interest of time, one burning question, if there is.
Yes, please.
Yes. Dr. Mugiria.
Thank you.
Thank you very much, Professor Ching Song. I pronounced it correct. Sorry, colleagues, I arrived when introductions had already been taken. My name is Roy Mugira. I'm the CEO of the National Biosafety Authority, the authority that is charged with the responsibility of regulating genetically modified organisms. Thank you again, Prof, for even hosting us last week at your lab at Michigan State University. I was in the delegation that is still there.
Yeah,
I had to come back to attend to certain other matters. I just want to update the map on GM crop adoption that was displayed of 2016. That space has changed. We now have, we are implementing BT cotton in Kenya.
Yeah.
So a number of countries have come on board from the African context: Nigeria, Malawi, Ghana, and several others. So this is just to mention to colleagues that the technology is growing. I will be saying a few more things later in my presentation later today, and therefore I don't want to take much time. But also to indicate that our genome editing regulations have followed the same path as the Indian also as the Argentina, where we check and say, is this ending up with a GMO, and therefore this goes out of the regulation of the biosafety framework? Is this one ending up with a genetically modified organism? This becomes of interest to us. So we are updating that, and thank you very much for your presentation. Asante.
Yeah, thank you very much for your comments. That's correct. Like the GM crop adoption map, that was from 2016. By the time AAS, after that, they stopped updating the information. You are right, yes. That's very exciting. Thank you. Yes, please.
Professor Xing, thank you very much for your elaborate presentation. I have a very quick question and observation. You gave us, in terms of the genetically modified plants all over the world, and you said the most trait is the trait of herbicide tolerance or herbicide resistance. And some opponents of this technology claim that the reason why that is the case is because the biotechnology industry are trying to support in a manner to use a lot of glyphosate in this sense. What would you say to this? Is that true or not true? And if not true, what would you say to this allegation? Thank you.
Yeah, I think I don't know how to answer this, but I'll give it a try. Okay, I heard this about this. In my class, every year I open the debate for the students to have a pro-GMO or con-GMO. Okay, this is always bring up as evidence. Like, okay, the herbicide resistance crops development because Monsanto want to make some money because they control the herbicide resistance. But look, one fact is Monsanto is not there yet. Right? So Monsanto is just like it's bankrupted and is bought by Bayer. So to answer your question is, I don't think maybe at the beginning it's a profit driven. Maybe that's a reason. But overall, if we see like the contribution of this GM either soybean, mostly Roundup is for soybean, right? It's like the contribution is huge. If you look at the production of the soybean year by year, without the growers, they like that because it saves a more. Nobody like to do like the weeding things under the the warm weather or the hot weathers, right? So basically, myself, my opinion is I don't think that is very, very true. It can be like used as a point for like debate, for argument, but I don't think that's very, very true. Okay, so as long as in my mind, as long as it can benefit the growers, the farmers, or the or the general public, that's what be a good product. Unless we see it has some very, very unexpected impact. Okay, that would be a concern. The concern mainly is like environmental concern or food safety concern. We don't have food safety concern. That's the bottom line. Environmental concerns is a concern, but even the traditional we spray a lot of pesticides or herbicides. The other if it's not Roundup, we have to go for the other herbicide, right? We still need to pay. So that's my understanding. Yeah. Thanks.
Thank you very much. I will go to Mulwa's lab and Kimurto to see how they pick those small things and implant them somewhere so that we can also use them in the physical materials so that we, ours would not be that very dangerous to human beings because they are non-living. But we learn because we want to borrow from techniques or from the biosciences to the physical what? Sciences. I think that is a gray area which I will learn from you guys. My time is over. I want to thank you. I want to hand over to the next chair of the session, and she will know how best to catch up with time.
Thank you very much. Let's applaud the chair yet again. Thank you. Thank you very much, Prof. Indeed, we are running very late, and we want to see that we find a mechanism of recouping the time that we have lost. But we now have a very important session that really introduces us to the International Center for Genetic Engineering and Biotechnology Regional Research Center that will be established here at Egerton University, and is the host really of this workshop. And so we really want to hear an introduction of how we got here, and also to get a roadmap to the establishment of the center. Since morning, the presentations we have had, they are giving us ideas of what we need to do, what we need to consider, even as we prepare to establish this center. And I like also the presentations in the sense that a lot of questions and a lot of statements have been made that opens our minds to this whole issue of genetic engineering, and more importantly, creates a space for us to openly discuss and debate. I like what Professor Song said, that he allows his students to debate. There are those who are pro and those who are against. And I think it is very important that even as we introduce the next presentation, that we can have that open discussion based on scientific evidence, why we are pro or why we are against, and therefore we can come to a consensus, hopefully, as we consider the evidence that is available. So I want to introduce Professor Omolo. He needs not much introduction for the people in this room, but for the sake of those who are joining us online, Professor Omolo is a professor of chemistry based here at Egerton University, and he has asked me not to say a lot about who he is because of time. So because he wants to spend the rest of the time really introducing the presentation. He's been a former dean of the Faculty of Science at Egerton University, and also a director in charge of marketing and resource mobilization at Egerton University. Professor Omolo.
Thank you very much, Director Research, Professor Mungai. All protocols observed. Allow me to straight away try to catch on time. The Director, you were told to catch on time. Now I'm the one left to ensure that that's done. As a lot has been said about the RRC, ICGEB RRC, how it came to be is what I would want to say. The VC talked about it. The Director, Anna Costi, also mentioned that. So I'll try to, as much as possible, to be brief. And this presentation is being done by Egerton University, prepared by a team that was set up by the Vice Chancellor in the interim, to do a little that can be done before the center kicks off. I think for the interest of those who are joining us online, that is the face of Egerton University. Our website is over there. You can read more about us. But if you find a chance to come, at least you will have, it is a serene environment, as you have been told. ICGEB is a community of research organization that covers the whole world. I think Professor Sonti mentioned about one component being in Trieste, the other one South Africa, and in India. Those presently are the ones that have the state-of-the-art laboratory, but in the recent past, there has been a move to form or establish regional research centers. Ours is the second one to be established in the world after the first one was established in China a few years ago. So the map still just shows the presence, the print, global print of the ICGEB in the world. If you tease out Africa, that now becomes the African region. And for the moment, that's what we are referring to as Regional Research Center, the one that is going to be established at Egerton University. So that's just a show of the region, the geographical region that the region should span. Those are the one with the deep blue color are member states for the ICGEB. And as it was mentioned earlier, Nakosti is the focal point for ICGEB, and those would be now forming member states that would be having stake in the center that will be here. These are key dates that are important for the establishment or how this RRC came to be. It was also mentioned some of this, but the most important date here is 1st of July, which marks the beginning of this center. So in the interest of time, let me move on. When we conceived this idea, as we responded to the call that came about, we ensured and argued and aligned the center with the national, regional, and global development. And it's not a surprise that we are here where we are. What is really in the books of ICGEB? What is a regional research center? It is a national research center, or they are part of, with independent governance, which is shared between the ICGEB host country and the ICGEB, the host country, and the member state. And the purpose is to contribute in the strengthening of research skills, knowledge, and capacities of the local scientific community. And I can't read all because much of that has also been picked by the previous speakers. When we were invited to respond to this call, we looked at what was the span, the coverage of things that ICGEB already is doing or they do in their setup. And at Egerton University, evaluating our strengths, we found, and it was fitting to make a call on the plant biology and biotechnology that fits well with the agricultural, the agriculture, which is the mainstay of the university, and bringing in agricultural technology was the point. And some of the key areas that we put forward that were evaluated and as a basis to establish the center, or that will be established as we move along, are the 6 areas that are on the screen that basically are supporting the food and nutritional security. By then, we were thinking about East African community, but then here we are. The task is here for us. The kind of things that the center will be doing, empowering staff, and picking the good graduate students. I think this is important for younger scientists that are on the system to know that there are opportunities. Postgraduate mentorship, and research capacity development and technical capacity, and many more that will be helping to bring or establish the capacity further. This had been said by Director ICGEB. These are standard programs that are supported by ICGEB that will be run also at the center. And they emphasize training, research, and technological transfer. Is this center really of significance? Yes, it is. And just to give an illustration, when we rolled out this workshop, when we requested people to register for it, the response was very enormous. And I believe it will translate to attendance or future engagement, because we have already picked the details to establish a database that will form a platform for engagement. If you look at the attendance by country, you get that impression on the screen. Of course, Kenya, I think because of proximity, we seem to be dominating, but at least the good news here is that we have penetrated, as the Vice Chancellor said, and at least we have reached those countries, particularly the ones that we have interest in. Another thing is that will get better when the formal engagements are established, the framework with the member states. when this goes forward. Maybe again, just a snapshot, when people are talking about area of specialization, what stands out clearly with their registration was biotechnology. That word, I think when I randomized the registration, area of specialization, the word that stood out, and you can see from there, this is computer generated. But again, that is just underscoring that I believe we are on the right track. And by gender, the representation is 6-4, with the male dominating, at least. I think that is a key information because inclusivity and diversity is a key issue. And when AAP was also funding us, one of the conditions was that we must demonstrate inclusivity and diversity. And we think that that is pointing towards the right direction. In terms of training, level of training, I wish to report again here that doing a summary by grouping or bulking the areas that members or people posted in, I know some of them varies within the region, but if you take the level of bachelor's, master's, and PhD, they were almost in one, no, they were the same, all contributing 33% each, and 1% were from diploma. What do we get from this? The interest is anchored on the right people, and particularly if you take the first two, master's and undergraduate, and bachelor's, that gives the impression that even the younger generation are more keen on this. And there is a pool that the center can bank on. On that first part of this presentation, what we must talk about is the transformative potential. I think that has come out in the early part of the morning. And the optimism that this workshop will set a good stage. And I wish to report to the audience that the Vice Chancellor, Egerton University, has already allocated a website for this center. It is still under construction. It will be a platform for much engagement, but there is already an email that is dedicated for communication. I thought that was important for the general audience to know so that we have this center moving forward. Roadmap is a bit detailed. And what I will pick about, this forms the components of the roadmap. It's a big document that we are continuing to work on. But what I need to share here is the vision. The vision, and we can get it, is to establish an internationally renowned center for the genetic engineering and biotechnology that serves as a regional hub for cutting-edge research, innovation, and collaboration. The mission is also there. Mandate of this center can be summarized as research, training, innovation, partnerships, and technology transfer. And that has been used to generate 6 goals. And within each goals, there are objectives that are set. The first objective, is, of course, is to bring the state-of-the-art research facility that meets international standards. And goal 2, all the same line. Goal 3 is on technology transfer and commercialization. In its life, that should be able to come about. But the roadmap in its 5-year plan, and as I'll show, we have that ambition we hope that it will achieve that. So that much is in the detail. We've further done a lot of current asset, we've done extensive current state assessment, and some of the inputs, again, we'll get from this workshop through engaging questionnaire that will be shared by the team with the members who are already on the platform. And we invite your comments so that we enrich that roadmap, okay? Key strategic initiatives that will be focused on: infrastructural development, research program development, talent acquisition and training. And this one, it will be clearly established defined by the steering committee that will be formed in the life of this, we believe, in the next few months. Collaboration, the issues of technology transfer. Again, more important, how to engage in stakeholders. And this center, as we have seen, the stakeholder spans a very large part of the map of Africa. And to just summarize, I believe maybe this is not clear, but all that I've mentioned up there is refined to at least now distribute all that information in a 5-year plan. I will not go into each of those, but it's just to demonstrate that there is a plan. We've done also risk map. This is something that always continues. And there is NNME plan, communication plan as well for that. And allow me just to move faster, just to move on and see the kind of structure that it will have. It will be an international center within the university, set up, the government of Kenya, but again shared with the international community. And all that information will be fully covered and shared on the website for the RRC. Allow me to stop there. Thank you very much. I hope I've caught the time. Thank you.
Yes, you've done very well. I think we are catching up on time. And at this time, we want to acknowledge the over 100 online participants and encourage that you continue to put your questions and comments on the chat, and we'll be picking these to share also in this platform. So at this point, really, I don't know if there's any reaction to what Professor Molo has done. Could give just one reaction before we take a break for a short break. All right. I think everybody now is clear and has appreciated the center that is going to be established yet. Okay, very well. Just one comment from the mic, please. Somebody can share the mic. For the sake of the online audience, yes. Mm-hmm.
Thanks, Professor Molo. For the interest, you chose to show us degree levels. I thought you would have shown us the interest by sector so that we know who are showing interest. Public, private, NGO. I think you have that information.
Very well. It's a question about the sectors that the participants are drawn from.
Thank you very much. We have that information, but because of time, we didn't analyze. But mostly universities, research institutions, and most of them were in the universities and research institutions. But that's not all. There are also others that are in non-research institutions. Like this is not in Africa, but there's a team from Iran. I believe they're online. They were very interested. They were bringing a very diverse kind of people that are interested. That information we are going to work on and then post it also on the website as a report for this workshop. Thank you.
Okay. Thank you very much. I know we haven't taken any question from the online audience, so allow us to respond to this one. There's a question asking, is the center only focused on plant biotechnology? And I think we kind of know the answer, but just kindly go ahead and respond to that.
We start from somewhere. We'll start with plant biotechnology. As we grow along, we'll evaluate and see on the strategic plan. Thank you.
Okay. Very well. So whoever asked that question, I think you have gotten your response. We start with plant biotechnology, and then we'll grow into other areas of biotechnology. So I'd like to invite us now to a group photo. Outside, for those who are here physically. For the online team, we're taking a break of about 20 minutes, and we'll be back after that. So we'll take a group photo, then a cup of tea. Thank you very much.
Yes, good morning. The next session is about establishing research groups and collaborations. We are going to have three presentations. The first one by Dr. Shams Yashani. The second one is by Dr. Bernard Kurohoma. Then we will be having Dr. Roy Mugira, the CEO NBA. So I believe that Dr. Shams Yasani is online.
Yes, I'm here.
Are you there? Okay, welcome. So I will give a brief introduction of Dr. Shams. Dr. Shams Yasani is the group leader of microbial engineering at the International Center for Genetic Engineering and Biotechnology, that is ICGEB, in New Delhi, India. He is a distinguished expert in the field with a strong academic background and extensive research experience. Dr. Yasani has made significant contributions to microbial genome editing and engineering, focusing mainly on biofuel production from non-food-based feedstock. His innovative work includes enhancing enzyme production for biomass degradation and engineering microorganisms for efficient ethanol and advanced fuel molecule production, showcasing his profound impact on sustainable biotechnology. Dr. Yasani, welcome.
Good morning. Can you hear me? Hello, can you hear me, man?
Yes, we can hear you. You can start your presentation.
Thank you. Thank
Yeah.
you very much.
Yeah.
I'll just share my presentation.
You can share his presentation. Oh, okay. He's sharing from the other side. Okay.
I'm sharing from here.
Okay, go ahead.
Can you see my slide? I hope you all can see my slide. Okay.
Yes, we can see your slides.
Thank you very much, first of all, for giving me the opportunity to speak on this very, very important occasion on the establishment of the ICGB RRC Center at the university in Kenya. I'm very, very excited about this development. And the topic that has been given to me about talking about establishing research groups and collaboration. I'll tell you some of the experience about what we have done at ICGB New Delhi on establishing some of the research groups here. And let me start with the statement that our director, Dr. Sonji, has made. You know, in India, we are working on this area of agricultural residue validation, mainly because there are issues. Those residues are produced in excess, in a much, much higher quantity that can be utilized favorably, and therefore they are mostly being burned. So there is a government mandate that prevention is better than cure. If we try to work on preventing polluting our climate, we are able to tackle a lot of diseases as well. So biofuel is of course one such molecule which is carbon neutral, not releasing any carbon dioxide in the environment. It is absorbed by the plant and it's a process to make biofuels, which can be used in the vehicle. And that's the reason why the government is doing a lot of emphasis on making renewable fuels. Most of the fuels that are commercially available in the market at this moment are first generation biofuels that are coming from edible sources like sucrose, starch, or vegetable oil. Those coming from sucrose, starch, they are basically arising out of microwave fermentation, and those which are basically biodiesel are transformation of oil. But these are basically what is called fourth generation biofuels, are mainly made in some of the countries like US and Brazil, who make use of cornstarch or sugarcane sugar to make ethanol, and there are some biodiesel which are made in European Union. Now, most of these first generation biofuels are coming from those of food source, and which is leading to a huge problem in terms of food versus fuel and therefore a country like India can't afford that, and we want to quickly move to the next generation of biofuels, which we call them as a second generation and third generation biofuel. Basically, in the second generation biofuel, the problem that happened in the first generation where edible part of the plant has been used, we don't want to use that in the second generation, so it's basically plant source, plant diet, but non-edible part of the plant. Basically, mostly the residues, agricultural residues. And also in the third generation biofuel, which are algae, so future basically, people suggest that second generation will become limited. So eventually we have to look at the plants, water sources where third generation biofuels could be made. So with these mandates in mind, Government of India indicated that to establish a center, we will support you to work on second generation and third generation biofuels. About a decade back, this is the proposal that came to ICTV. ICTV made a proposal that, okay, we will establish a data center, a Government of India funded center, which will work on second generation and third generation biofuel. So basically, when I say second generation biofuel, it was a lignocellulosic based biofuels where the lignocellulosic plant needs to be deconstructed or enzymatically to make sugars, and those sugars will need to be fermented, both C5 and C6 sugar, and then fuels are made. Ethanol is a major fuel, but there are other advanced fuels which are much closer to the PhD in fermentation technology around 23 years back. So the first 10 years at ICTD, I was actually working on their vaccine development. But with the needs of an hour and the requirement from the government, I shifted to clean energy area, and I started developing a group which could work on enzymes, microbial engineering, and fermentation. We have a group leader, Dr. Sigurd, who worked on eastward for biology engineering and making of these biofuels. We have a systems biology person, Dr. Shri Srivastava, who works on different model so that we can get a target for information. We have Dr. Jitu, who is expert in NGX analysis, who makes various functional genomes to produce some valuable chemicals and commodities. And we have Dr. Shashi Kumar, who is known for his expertise in plant molecular biology. He shifted his focus on chloroplast and genome engineering, albeit to make biodiesel and biofuel. So basically, we had developed a core expertise at ICTD to work on second generation and third generation biofuels, and there are some overlapping in that could serve to both end of the biofuel program. So with this expertise in all domains we started working on, I'm going to tell you some of the work that my group has been doing. So basically, the lignocellulosic biomass is composed of lignin, hemicellulose, and cellulose, which needed a thermochemical treatment, which is really a chemical engineering challenge. So we decided to actually not establish this particular first part at ICTD. We decided to collaborate and act with one decision that we made in the beginning to develop a collaboration with some companies or with the Chemical Engineering Institute. They could have a different treatment biomass made available to us. We actually focused on developing enzymes because enzymes was very expensive. Dr. Nassim Wadley helped in developing yeast for C5C6 fermentation. My group also focused on other advanced biofuels like butanol, oxygen, alcohol, and ethanol, because they were really a drop-in fuels. So in terms of enzymes, it was difficult because those structural biomass, which are lignocellulosic biomass, are very difficult to hydrolyze. You need really a mixture of different enzymes. Just one enzyme is not sufficient. There are 4 enzymes, auxiliary enzymes. Sometimes even hundreds of enzymes are not sufficient to hydrolyze lignocellulosic biomass much more efficiently. So we started looking at hosts that could give us some good enzymes. We realized that there are not much people in the market who could sell enzymes for biomass hydrolysis. There's only one multinational company like Novozyme that can sell enzymes for lignocellulosic biomass. So of course there was a need of an hour to develop indigenous enzyme source and great form so that these could be produced in tons and used for the biofuel kind of work. So we collaborated with some of the plant entomologists at ICTD to work on the gut insect gut microbiome, because insects expel from those microbes to hydrolyze lignocellulosic biomass. So we screened those gut microbes. We also looked at different fungi, because fungi are excellent recycler of decaying materials. We looked at those as well. And we had, with the help of government, we could establish a very high-end facility to do high throughput liquid screening liquid handling facility so that we can do a high throughput screening. We also had a very good mass spectrometry which could actually help us to identify the nature of the enzymes that are being produced. So we basically made a small metabolic model to screen hundreds and thousands of enzyme sources that were generated by different bacterial and fungal hosts. And we eventually selected one of them which was being best result in terms of the biomass hydrolysis as well as in terms of the enzyme activity. And we compared it with the enzymes that were available in the market, and it looks like it was very, very effective enzyme. We selected that and we started studying both genomics and proteomics, as well as looking at some of the enzymes that were key in the secretome of those fungi. We characterized that. These are some of the proteomics results. And we eventually developed the tools to engineer the host, because these are natural isolators. Fungi that we selected actually were resistant to many antibiotics that was having problem in the transformation. We developed tools, including medicine tools, to transform that so that we can make it even better host for enzyme production. So we started looking at how to enhance its capability to produce more enzyme. We deleted the precursor. We can see that deleting the precursor can improve the enzyme production. We also overexpressed some of the key cell-lytic enzymes in the genome, like oxygenase and cell biometric, that eventually increase the capability and efficiency of the enzyme produced by the host. We finally showed that the enzymes that we are able to produce now could compete with the best enzyme in the market and can hydrolyze at a very good efficiency with biomass, and convince Government of India to fund us so that we can scale up our technology. So with the help of support from Government of India, we had CP and we scaled it up, the enzyme technology, fungal enzyme technology, to 15,000 liter scale, and we validated it, this technology, and it seems to be a very, very competitive. We took help of Institute of Technology, but to do some techno-economic analysis. Again, the cost seems to be very, very competitive with the commercial enzymes, indicating that it's a very good enzyme composition that could be taken forward for commercialization. So we received a lot of interest from some companies who wants to take it forward, and we're working with them so that we can further scale it up. In fact, this expertise also led to a very good collaborative project from India and UK. It all came from a cause where they wanted to make the waste coming out of the industry utilized for other purposes. And this sugar industry, as you can see, is a huge amount of sugarcane bagasse that are being accumulated as waste. We decided to work on valorizing this, and there was a huge collaborative partners involved in this, both from India and UK, University of Geography, one of a number of companies that were involved. That really led to a very, very effective enzyme composition for sugarcane bagasse hydrolysis that came out of this technology. It's also very, very promising, and we're trying to see how we can use it for valorizing sugarcane industry's waste. In addition to those enzymes that I've shown, we are also working on some of the advanced fuel molecules like butanol. This also is coming out of a very good collaboration with a US partner, Professor Matthew Kofas, very good synthetic biologist, and that has led to a new pathways for production of butanol. These are some of the details we can discuss if people are interested. The last part of my talk is on hydrocarbon production. So, basically the petrol or diesel are hydrocarbon. There is no functional group attached with those hydrocarbons. So if we can produce hydrocarbon directly from a source, it is a perfect petrol or diesel. It's not really a biofuel or biodiesel, it's a diesel. So that's what we try to do. We actually found a pathway which could be engineered in bacteria, and that could be used for producing C15, C17 chain alkane, which could be used as a drop-in fuel. Also, it is a paraffin which is of interest to the cosmetic industry. We also, in the similar pathway, produce long-chain alcohol called citral alcohol, which is composed, which is also a composition of facial cream. So that also, in addition to fuel, has an interest in the cosmetic industry. So we basically look at these pathways, we engineer it, we of many industries. They came forward and they wanted to do some collaboration so that they can together produce some of the important commercially valuable product. For example, Hindustan Petroleum is a petrochemical company in India. They want us to produce cellulose enzyme for their own specific purpose. There's another company, Biotechnology, another petrochemical. They want to produce a platform chemical, one for butane diol that can be used for making rubber. And there's another company, Egerton, which has funded us to work on this collaborative work so that we can use fungal platform for making milk protein. And with our some of these cosmetic products, we have now new discussion started with L'Oreal so that we can see if those cosmetic, microbially produced cosmetic product can further be utilized for commercialization. So with this, all credit goes to the students who work on this and collaborators and funding agencies. I would like to thank you and back to you.
Thank you, Dr. Yassani. Basically, he has presented on the wide biotechnology, talking about the industrial production of chemicals. So we could like to request if there are comments or questions, maybe two. Yes, go ahead. Do we have a microphone?
It's not working. Okay, thank you. It's working. Thank you for the very current and exciting research that you are doing at the ICGB Center. Just mind-blowing as I was following through, especially its relevance in transforming waste into biofuels. And we will assist in the current issue you're having with climate change. I saw one of your collaborators, and that's a question I'm trying to ask is, who was Dr. Pavan, who's majorly on NGS and omics. So one of the challenges which we learned earlier is that setting up downstream processing for ICGB facilities is quite costly. So do you outsource these services, or do you have an in-house NGS and an omics facility in your center? Thank you.
Thank you very much for your question. Very, very important question. So we don't have a genome and transcriptome sequencing facility at ICGB, so we outsource because we found it is much cheaper to outsource rather than establishing those facilities at ICGB itself. But we do have a very, very good and sensitive proteomic facility. So the mass spectrometric facility that we have is very, very sensitive because we realize that for our deep research, it's better to have that facility in-house. That would lead to a better invention.
Okay. So downstream processes, they also outsource? I think you asked about the downstream.
Yes, the downstream analysis, which is part of the omics. Once you have your data, now the NGS analysis, the proteomics and metabolomics, and I think it's good you've mentioned the proteomics facility, which is which also cuts across organisms.
Okay.
Thank you.
One more question. Yes, Prof.
Thank you for bringing us to the state-of-the-art technology. I recognize that it's very expensive. In here, we want to be where you are. Please comment on how you mobilize your resources, how much comes from outside the government funding to you, and who are these?
Okay, again, thank you for a very important question. So I must say that the major infrastructure actually came from the government support through the spatial program-based support on the clean energy. But the research funding we generate from different sources, both equally well from private as well as from the government organization.
So, okay, let us appreciate Dr. Yassani.
Thank you very much. Today I'm actually in Malaysia attending a ICGB workshop, so it was really a pleasure virtually connecting with all of you. Thank you very much.
Thank you. So our next presenter is Dr. Bernard Kurohoma. I hope Dr. Kurohoma, you are online.
Yes, good afternoon, everyone.
Okay, good afternoon. Let us give a brief introduction of Dr. Kurohoma. He's going to present on the experiences from the International AIDS Vaccine Initiative, which is strengthening capacity for HIV vaccine and biomedical prevention. Dr. Bernard Kurohoma is a Director of Organizational Capacity Development at the International AIDS Vaccine Initiative in Kenya. He's also a distinguished professional with extensive experience in genomics, bioinformatics, and capacity building. He holds a PhD in genomics and a master's degree in bioinformatics. Dr. Kurohoma has led significant research initiatives and has served as a principal investigator and a senior lecturer at esteemed institutions like the University of Nairobi and the University of Oxford. His expertise spans infectious diseases, non-communicable diseases, and population genetics, making him a valuable contributor to the workshop's discussions on organizational capacity enhancement for sustainable development. Dr. Kurohoma.
Thanks a lot. Please allow me to share the slides.
Is able to share? Yeah, you can be able to share now.
Thanks a lot. So do let me know if you're able to see my slides.
Yes, we can see.
Right. So my talk is on the other end from Dr. Sham's. So it focuses on the people. So the session is on the systems and structures that we need for sustainability. My focus today would be on the human resource. So I'll be talking about strengthening capacity for HIV R&D, drawing from the experience from our advanced program. So I'd like to acknowledge our funders who make our work possible. So my talk will include a brief introduction of IRB, which is our work, and the advanced program, and then I'll have a deep dive on the leadership development program, which would enable you to perhaps draw examples of how to structure implementation of the new program. So I work at IRB, which is the International AIDS Vaccine Initiative, and it's a non-profit scientific research organization that develops vaccines and antibody products for various diseases, so tuberculosis, HIV, and we are also working in the emerging infectious diseases space where we conduct clinical trials for Lassa fever in West Africa, so Sierra Leone and Nigeria, as well as development of products for preventing Marburg and Ebola Sudan. So our mission is to translate scientific discoveries into affordable and globally accessible public health solutions. So there are multiple programs for which address different areas of emphasis of these diseases, and the one that speaks to HIV research is the ADVANCE program. So ADVANCE is an acronym for Accelerated Development of Vaccine and Immune Technologies to Combat the AIDS epidemic. It's a 10-year work that we have in the USA through PEPFAR, And we have two main objectives. The one is to conduct preclinical and clinical research for vaccine R&D in the HIV space. And the second one is to strengthen the associated capacity for this. So in terms of how we're structured, we have four main strategic areas going from our objectives. The first one is the preclinical strategy area, where we do most of our scientific discoveries for the HIV vaccines and products that we develop. The second one is the clinical development department, which involves the conduct of innovative HIV clinical trials, as well as sentinel epidemiology trials in that space. The third strategic area is the capacity strengthening strategy area, which is going to be the focus of our talk today. And the fourth one is the translational aspect, which entails moving the discoveries that we have into products and making this accessible for the communities that we work with. So our work has a long history, and we work in several areas. So here I'm showing the main areas where we work in the global south, which is in Eastern and Southern Africa, and also with partner institutes in India as well. Also just to mention that we have labs in the global north, in California, a hub in New York, one in Amsterdam, and one in London, in the UK. So going back to the third strategic area, and I would like to describe our approach to strengthening capacity. So we look at it at three tiers, which is the individual, institutional, and regional levels of strengthening. And this is informed by continuous feedback. So it's a very participatory process that enables us to understand the evolving priorities of our partner network. So there are multiple programs that aim to ensure we address all this. Today I'm going to be speaking to an individual level, but also just mention that there are other levels. So we have international training program where we provide fellowships for people with youth experiences, which enables us to address implementation science key questions. Half of the population of the world, 4 billion, is women, and so we enable women to take leadership in research, as well as changing their environment or settings in which they work in, even as they have worked hard to change their barriers in their mindset. The other beam of work is the research management, which ensures that we have robust procurement supply chain systems. We have financial audit, compliance, we spoke about work planning and reporting, as well as human resource processes. Would also enable strengthening of scientific communication. And recording in progress. We would like to really influence and set a research agenda not only on the continent but beyond. So here we have a trusted research environment that enables us to not only safeguard the data assets that we have, but also share those out with the wider community. A biorepository that enables us to revisit valuable samples that we've collected to address key questions that may arise. A strong arm for South-South collaboration that enables exchange of best practice, as well as working in partnership with other key institutions in the region like Africa, CDC, and ABMI to discuss issues to do with vaccine manufacturing. So back to the leadership development program, which is what I was requested to speak about today. So the goal of this is to develop a critical mass of promising leaders within the advanced partner network who are able to advance HIV vaccine R&D in India and Africa. So that it aims to ensure that we have a well-rounded introduction into HIV R&D, provide broad expertise to effectively manage our diverse range of research portfolios. So for example, immunologists, we should essentially be able to manage a center that is working in community engagement or genomics. It also provides upskilling in technical and soft skills, as well as providing grant writing and niche support. So essentially, we're trying to nurture the next generation of leaders, and this is strategic because it helps us to formalize our training so that it's very standard across our network. It positions the scientists that we have playing more meaningful roles, and it's also strategic in terms of succession planning within our network. So what progress have we made? We started our conceptualization in 2021, which was very participatory, involving the different leaders at this clinical research center partner network. And it enabled us to identify the barriers that are faced by scientists within our network. So we prioritized the areas that we provide support on based on these findings. So just to speak to some of the cohorts that we have, as I mentioned, the leads or the leaders of the centers actually enable us to identify people who could be planned to take on more leadership roles. We pay close attention to having gender balance and also have a representation of the different areas of research emphasis. And this is across the 6 countries that we're working on. So as we mentioned, we try and do this using a robust approach, which is recording in progress in this case. So this helps us to integrate the technical and soft skill training. We also encourage participants to have an individual career plan, which they receive mentorship support to have fidelity to, as well as grant writing training support. And as you can see at the bottom in blue, this is for a diverse range of research areas. So looking at the international lead experts who we work with, they come from both the global north and global south, so the distribution is fair between these two regions of the world, with a slight disproportion of female lead scientists in our lead experts cohort, but with equal distribution from the different areas of research emphasis and markedly from the basic and clinical science space. So this is my last slide, and I think people are really important and they form part of the organization, so we can plan to take on new well-designed innovative studies and actually seek out extramural funding for them. But this is actually accomplished and led through the human capital that we have. So mentorship is really important, and we take it seriously by making it very structured for both our mentors and mentees at the beginning. And we try and make it work in such a way that people have a best fit. So if it doesn't work, it really doesn't work, and we really try and have people pulling others to the next level where they think they could work together. And the process is really mentee driven, and we usually have check-in meetings just to see how this is going. So I'd like to thank you for listening to me and for having me share this aspect of research management and how to strengthen systems, looking at the aspect of the human resource that you have for Prospective Research Center. And I'd be happy to take any questions at this point. Thanks.
Thank you, Dr. Kurohoma. I think from your presentation about capacity building, what I have captured is about technical skills, soft skills, mentorship, leadership training, all that. So I will want to have two burning questions if we have. Our online, do we have online questions from our online participants? No question. Now from the audience, any comment or question for Dr. Kurohoma? Yes, Prof.
Thank you, Bernard, for that presentation. I would always wish to listen to it always. I remember we listened to it early this year.
Oh, okay.
And from your experience, what would you advise in terms of research groups in a new setup like this to start with? Thank you.
I think there's usefulness in having quite good and diverse groups. I would imagine from these platforms, obviously with time, there would be refinement to actually see the specific areas or direction that you intend to take the research for this new center. But then it would be nice to have leaders who are able to not only integrate their research across teams, so not have silos, but also have a broader vision that we're all working towards certain goals. So I think perhaps we should all end towards the arc of accomplishing the SDGs, as the previous speaker has said.
Thank you, Dr. Kurohoma. We appreciate, and we look forward for consultations. Now I want us to move to the next speaker. The next speaker is Dr. Roy Mugira. He is the CEO, National Biosafety Authority, and he is going to present on the ethical considerations and the regulatory compliance. Dr. Mugira is an accomplished molecular plant virologist, combining his proficiency in molecular diagnostics of plant viruses with genetic modification for functional genomics. His role as the acting technical director and the CEO of Kenya's National Biosafety Authority attests to his expertise in biosafety and regulatory compliance in genetic engineering and biotechnology research. Dr. Mugira's substantial contributions to ethical considerations and his involvement in international platforms like COP, MOP 5 and 7 makes him a valuable participant, enriching discussions on biosafety, ethics, and genetic engineering during this workshop. Welcome, Dr.
Thank you very much, Dr. Nduko, for that introduction. I will start as the system is coming alive by appreciating the invitation. Thank you, Professor Omolo, and the organizers of this workshop. I struggled to find to be able to come physically because I really wanted to be here. Now, I changed the topic of our discussion today, likely, so that it becomes more relevant to what we are putting up in this conversation. And I want to outline my objectives right from the start. I came with 3 objectives. Number 1 is to make a personal and institutional commitment to support the establishment and growth of the ICGBE Egerton University Regional Research Center, both personal and institutional. So as an institution, as the National Biosafety Authority, we were behind this. We started the conversation when Professor Omolo, Professor Mulwa, and Ogendo visited us at Nakosti. That time I was serving as the Director of Scheduled Sciences at Nakosti, and that is where the conversation began. So this is really personal. I want to follow this through, and I want to see that this particular center takes shape and we're going to support it with all what that it takes. The second objective is to come home to my alma mater. I'm a double alumni of Egerton University, having studied my BSc and MSc here. So when I look around, I see familiarity in terms of where I am and in terms of people that I can see. Some are my classmates, like Professor Kimurto. We started MSc program here together. Professor Mulwa, Professor Nancy Mungai. Nancy Mungai was the first person who gave me a job. She made me sign a contract to help maintain the plants in CMRT. When that time working with then Dr. Ken Obura and Professor Ogendo there. So therefore it feels home. I also met Jane Rose, who was serving us in the lab, and I can see the community has grown. At the photo session, I met Bwana Bebo, the legendary photographer, who helped in making photographs of my slides and my petri dishes as I was preparing to do the defense. Professor Duda, we were at the Academy of Sciences together. Professor Ogweno, we went to a school in China. When I arrived in China, I found Ogweno and Professor Opio. I was struggling with the language and with a lot of trouble. When he said, "Abari Bwana," I said, "Oh, now I'm home. I'm home." I felt really at home, and many others. I see Professor Bebe, I love the Dairy Place, and one other, I think now Professor or Dr. Steve. We just used to call him Steve across there. We found him as a lab technician, and now he's somewhere else. So that is objective number 2, to come back home and meet all these guys that I've mentioned and many others. The objective number 3 is to make the presentation whose key message, and this is really to the research community here and those who are following us online, is that the Kenya Biosafety Regulatory Framework, which I manage, is sufficiently robust to assure safety and facilitate innovation. I remember I said not to facilitate research, to facilitate innovation, so that that which comes out of research can find its way to the commercial platform and therefore generate income and become something that is of benefit. It becomes a very difficult balancing act. You heard the debate that Professor Song makes in his lab, pro and against. As a regulator, a lot of times I have been accused of being a promoter. But it is a very thin line. If I have to teach and educate people about a GMO, I must say what it is designed to do, what problems it is overcoming. When I take that route, people start pointing out. People starting, is that comment meant for us? Somebody can check and see whether it's meant for us, or is a mic that has gone up there. Good. So, but I have a responsibility to see that which you do in the labs finds its way to the commercial arena, but also safeguarding safety. So I'll go through very quickly what we do and what we do to assure safety, because some of these things are things that you do. One of our reviewers is seated here, is actually our facilitator, Dr. Duko. Is one of our expert reviewers. So when we receive this, we don't do it ourselves. We receive your applications and pass it to these experts. They give us feedback. So I want to move very quickly to look at our regulatory framework. And the message is, how do we balance innovation so that we can assure that you do what you do, but also at the same time regulate to assure safety. Somebody will need to assist with moving the slides. Uh-huh. It worked, Paul. Thank you. Possibly found that you're on your way here and decided to behave. Some bar is blocking something. But basically, I want to start where the conversation began. When it became possible to move genes from one organism to another, and therefore crossing species barrier, the international community came together and asked questions about safety. So along that, they discussed and agreed on a protocol called the Cartagena Protocol on Biosafety under the Convention on Biological Diversity. In short, therefore, this is the global governance platform for genetically modified organisms, also called living modified organisms. As a country, we signed this in 2000 and ratified it in 2003, a clear demonstration of Kenya's commitment to the global governance of modern biotechnology and an expression of aspiration to become a key participant in the global biotechnology enterprise. As you have seen from previous presentations, biotech is leading the development in this particular area, and therefore we want to become a key participant. And this expression is picked from Kenya's policy. So the policy says, in 2006, we intended to become a participant in the enterprise within 10 years, and we are following through with that. The slides are taking a little time, but I think... Good, thank you. So after the global community came up with that global framework as a country, in 2006, we developed the Biosafety Development Policy, which gave birth to the Biosafety Act, which established the Biosafety Authority. That is the institution that I had. And after that, we did the 4 regulations. The first one is the biosafety contained use. The contained use element is what basically would be happening here within the labs, within the greenhouses, within the growth chambers, an area that is completely contained, even the confined field trials. The export, import, and transit, these are regulations that will say what you need to do if you intend to import, to transit, or to export genetically modified organisms. The third regulation is on environmental release. This is where we are setting it out to grow in the farms and to be commercialized. And finally, the biosafety labeling regulations. This is not about safety. It is about consumer preference. There is somebody who will say, I don't care whether it is safe or not, I just don't want it. So there is need to say this is and this is not GMO for purposes of facilitating choice. We work with a number of agencies. I don't know whether there is a bar that is blocking part of my slides. Please help me to put this somewhere. Good. Or we can just leave it. Okay, we will proceed with that. We'll see how to manage that. I need to go back. We work with 8 other regulatory agencies to be able to govern the whole spectrum of biotechnology safety. Number 1 is the Department of Public Health, because of course questions have been asked on the health, on the safety to human health. Number 2 is the Department of Veterinary Services. This one again focusing on animal health. The Bureau of Standards, the Kenya Plant Health Inspectorate Service, KIPI for intellectual property. Remember, this is a technology that carries proprietary rights, and KWS and PCPB. What I want to emphasize here is that within institutions like universities, we have institutional biosafety committees, And that is what we want to emphasize and to support Egerton University to set up the Institutional Biosafety Committee, which is the first filter of the work that you do before it comes to us. And once you have done that, when it comes to us, it becomes very easy, because you, as the experts, you have already filtered to check what is the research is doing. So what have we done over the years? Environmental release approvals. We have released these 3 crops, like I was updating the map with Professor Song. One, BT cotton is already growing with farmers. We approved this in 2020, and that is when it was commercialized. We are going to the 5th cycle of cultivation of BT cotton. Farmers are speaking about it, and BT basically is derived from the name Bacillus thuringiensis, the bacteria from which the gene was obtained to confer insect resistance to this particular cotton variety. The other one is BT maize, the same technology now in maize to overcome Africa stalk borer, and this is waiting variety release. We had concluded everything in terms of checking safety, and we were just about to go to cabinet. Remember, there was a ban on GM food, which I'll talk about shortly. So when we were preparing that, the government lifted the ban on GM food, and then we started preparing now to release this to farmers. Then some people went to court. Again, the activism that is out there. The judge gave orders stopping us from importing, from cultivating, and also from distributing. So we are battling it out in court, and the matter is out there. The third crop is virus-resistant cassava for Africa, which is a virus-resistant cassava developed under the technology that Professor Song explained here a bit on RNA, RNA interference or gene silencing. This is now going through NPTs, National Performance Trials. It is already out of the pipeline of safety. It is now being checked how it is performing out there. It is led by a team from Caro and the University of Nairobi. So what we do when we are approving a confined field trial, which is a category of contained use. First, an applicant, Egerton University, Caro, Nairobi University, whoever, submits an application, which is checked at the Secretariat of NBA, and then it is sent to experts and the relevant regulatory agency. If it is a crop, sent to KEFIS. If it is a livestock, sent to DVS, and then these ones give us feedback. The feedback is compiled by ourselves in the Secretariat, submitted to a committee of the board of NBA, which now makes recommendations to the full board, and the full board says, yes, this is approved, or it is not approved with reasons. Usually here we want to encourage research, and therefore all what we check is the adequacy of containment and handling. Are the people handling the material properly trained to do that? When making decisions for biosafety approval for environmental release, the process is a little longer and involves a little more people. And this is what I want to highlight, because it is basically the same as the one for contained, but when you look at the area of technical review, you will see up there public notice. We publish the application in 3 widely circulated newspapers for the public to give their comments. This is a requirement. Remember, we did this law in 2009, and this law was forward-looking, giving provision for public participation even before the Constitution came in to make it heavily mandatory. So we send it out to people, to the newspapers, and give portal where they can make comments, even through text messages. The other thing that you see, which is different from the one for contained use, is the experts. Among them, you can see some point here. We have SECs. These are socioeconomic experts. We also assess the socioeconomic implications of the project when it gets to the environment. And then again, we send to the relevant regulatory agencies, KEFIS, for example, NEMA. Especially NEMA, we must involve NEMA because we are taking it to the environment. And then the scientific community who submit their submissions, including the public submissions, the socioeconomists, the regulatory agencies, the experts. This is synthesized again, presented to the committee of the board, to the full board, and approval is given or not given, and then it goes to NEMA for environmental impact assessment, KEFIS for national performance trials, and eventual release to farmers if it is a crop. So far, we have dealt with these numbers. I think, sorry, my table needs to be updated. These are figures of a while back. In contained use, I think we are now at 42, and with this can be updated. But generally, we have handled these applications, and the approvals have been given. Contained use, confined field trials specifically, we have handled 15. Import transit before the ban, we handled 28. 2 are pending because of the issues that I've mentioned before court. Environmental release and placing on the market, we have approved 3. 1 was rejected. And I want to emphasize that which was rejected was a flower called Gypsophila. Gypsophila is a flower that is used to fill in flower bouquets. When one is making a bouquet of roses, you fill in with some white, small white flowers. Now, the international market has been craving for a blue Gypsophila. Gypsophila normally is white. So there is one which is genetically modified to produce blue flowers. A company came to Kenya in 2018 to ask to do trials. And that was rejected, not on the basis of safety, but on the basis of socioeconomics. There was an argument that if we adopt a GM Gypsophila, it will affect our flower market to Europe. Remember, Europe is still jittery about GMO technology and genome editing. So because of that, that proposal application was rejected. They appealed, and it was discussed again, and the board sustained the rejection. So, but when you come to ask it, we can find out whether this can be revived, because if we are modifying Gypsophila, it has no effect on roses. So there is no reason to stop our roses getting to Amsterdam if we haven't modified roses and Amsterdam doesn't want genomes. Sometimes a lot of the discussions that we hear is commercial turned political and mixed with science and religion, and then it becomes a potent argument that you don't even want to engage in. Now, fast forward, 2012, the government banned the import and use of genetically modified organisms. It was based by an infamous publication by Professor Cellarini and others. This publication was publicized even in media and newspapers, portraying rats as having developed tumors after feeding on GM maize. Now, later on it was disapproved. In the country, the ban was imposed, a task force was formed, but those of you who might have followed this, the science was found to be completely flawed. I think it was some of the science somebody can be asked to do to prove something. Because first of all, the mice that these scientists used are genetically bred to develop tumors. So whether they feed on water, whether they are starved, whether they stay, they will develop tumors, because they are genetically bred to develop tumors to assist scientists to study how cancer develops. So that is really, really a flaw. You can't use that which you will develop anyway. Then it didn't have the proper controls. You scientists, you know that. Until the publication was pulled down from the journal that initially published it. Of course, your scholars, you know what that means. If your work is pulled down after being found to be not to meet certain standards. So in 2022, the government lifted the ban following our advisory. Fortunately, we have a president who understands science. So when he asked us to explain a few things to him, he was convinced that really there is no scientific basis, and out of engagement with the cabinet, the ban was lifted. Then we started now picking our regulations from the shelf. Remember, we made regulations in 2012, and then a ban was imposed. So we took our regulations and our law and our guidelines and put them in a freezer. And the institution almost died. Because if I establish you to regulate this water that we are drinking, then I ban the drinking of water. Where do I leave you? Or if I establish you as a university to teach, then I ban teaching. You are left. So For the last 10 years, we were in the freezer. We came out of it, and we were very excited. We started saying, now we found some work to do, and we developed guidelines on how to import. Then quickly, those guys on the streets went to court. It has also been turned political. In fact, the first petitioner who went to court is one Mr. Mwangi, who is also a lawyer that is associated with certain side of politics in the country, and therefore went quickly and put a petition before court. Some other groups came in, and the judge made a ruling, made an order. Those orders that are called early orders. And we were asking now, she made those orders in what lawyers call ex parte. Ex parte is when orders are made without you. So she made it without us. We went to the Court of Appeal. We were thrown out. We tried to plead with her. She refused. We tried to ask to be given 3 judges, and it didn't work. We are now going for the full hearing, and we shall be assessing this. Again, the court also allocating itself. I can't comment on that much, but we are handling those matters in court. So that is where we are. Even the maize that was ready, with Caro, with some seed to come from some place to give to farmers. Now we are holding on to it because we have been told to stop. But we are still doing cotton because that was approved earlier. But I think that is also a legal argument that we need to understand. Now science keeps evolving, particularly this field of modern biotechnology. This was presented earlier by Prof. And now what we're talking about is genome editing. Instead of bringing a new gene from another source, you get into the constitutive genome of the organism and edit in the same way that you would do a paragraph and change the meaning of the text. So these 2 ladies, Emanuela and Jennifer, won the 2020 Nobel Prize for developing the method for genome editing, which was followed again later by the professor from MIT. Now, this has become more favorable. It is easier to manage. In fact, you can do this using what they use now in TikTok called do it yourself, DIY. If you get the right enzymes and right things to do, and it is less regulated, as I'm going to show in a short while. So when you perform this, there are 3 possible pathways, SN1, SN2, SN3. One way gives you a GMO at the end of the pipeline. So that becomes of our interest as an authority. The other pathway, you start with a GMO because you have been brought in some genes to help you do some other things. But when it is exiting the product stage, it is non-GMO. The novel gene has been taken out. So it becomes of our interest as biosafety authority up to the point when it exits the status of GMO, particularly within the confined area. When it becomes a product and it isn't carrying any novel gene from an outside source, then we release it to CAFES, to DVS, to whoever else now regulates other aspects of crop and plant and animal development. The other pathway is completely from start to end, no GMO interaction, and that becomes really not of our interests. And since genome editing is not covered under Cartagena Protocol, or even under our laws, we started asking ourselves, how do we regulate this? And that is why I said my key message is that our regulatory framework is sufficiently robust to facilitate safety, to ensure safety and facilitate innovation. So we went back and did guidelines. Of course, learning from other parts of the world. We learned largely from Argentina and from others who had done ahead of us. So we developed, therefore, a guideline that provides a technical guidance to applicants on the criteria that we use to determine is this one GMO, is therefore subject to our regulation, or is it not GMO, and therefore should go to the next level regulation. So we developed this, and the key features of these guidelines, number 1 is an early consultation stage. So we don't ask you to do a dossier. Dr. Nduko is familiar with what a dossier is. A dossier is a huge document that the applicant puts together to show this project has been checked and so on. The first thing we ask is a very simple form. Tell us how you made this genome editing. Then our people, sometimes we may consult experts like him, tell us, is it ending up with GMO? Very quickly, within that days, that is bullet number 2, we are able to say, This one will follow this pathway. This will follow this pathway up to this point, and therefore we can be expeditious in our flow. We have a clear flow of approach, and this approach has been adopted by many other countries. Incidentally, UK has published a law that has learned from this. I was very happy when somebody called me and said, you see, Roy, your guidelines have informed law in UK, and said that our former colonizers are now learning from us. And I took credit for that. A number of other countries are following this method, this approach: India, Argentina, Nigeria, ourselves, and so on. And the purpose, like I said, colleagues, is to make sure that your space of innovation goes on as we put an eye to check on the safety. So this is the flow chart. Basically, we start early consultation with NBA, you fill in a simple form, then we check for completeness, the same line. Is there integration of foreign DNA sequence? If the answer is no, then that is an non-GM, and we pass it on to the downstream regulators. If the answer is yes, is the foreign DNA sequence present in the end product? If the answer is no, again, we revert to non-GM and pass it to the downstream regulators. If it is present in the end product, then the answer is yes, we notify the applicant and inform the applicant that this one will have to go through the risk assessment process as a GMO. We have approved, I don't want to use the word approved because this is not within our purview, but we have dealt with these 3 applications so far for genome editing. Number 1 is maize that is edited for maize lethal necrosis resistance, the virus that has a combination of viruses that has dissipated maize production in parts of this country. This is led by a team from the University of Nairobi and others and partners from MSU as well. Professor Douglas Miano of Kabete campus is handling this. And the essence, therefore, is to have a maize line that is resistant to MLN using genome editing. The second one is genome-edited nitrogen-fixing bacteria used as a seed treatment for cereal crops. Bacteria that fixes nitrogen traditionally associates with the bean family, the legumes. Now, this one is genetically edited to associate with maize, sorghum, sugarcane, and therefore it is able to assist these plants to fix nitrogen and reduce the amount of N-top dressing required. It is already a product in the market by a company called Pivot, which is in the US. And these 2 crops are now undergoing what is called national performance trials. Because from our side, we checked and saw that they do not fall within our regulatory frame, and we have passed these to KEFIS. The third one is genome-edited sorghum lines with resistance to striga. Of course, you know striga and how it causes trouble with sorghum. This is being managed by Professor Runo of Kenyatta University, together with the Koteva and some other private sector entities. And these sorghum lines are undergoing, again, national performance trials, since we said, after assessing, the end product is not ending up with a GMO, and therefore it can be regulated by KEFIS, as I've indicated here. Sorry, this bar is coming here. That these are now being regulated by KEFIS as conventional, because we have assessed and seen that it doesn't carry GM material. Good. These are my concluding points. It's okay, it's okay. I'll manage. Number 1, like I said earlier, is to pass a message that our policies and legal regulatory framework, an enabling one, is critical. If you guys are handicapped by regulation, then, and remember, sometimes when you do research, especially you that are teachers, you are, also passing knowledge. You are the forefront of knowledge generation. And therefore, some things you try may not even work. So if we stop you before even your MSc students can try out some things, Shall we ever have innovation? So the framework must be enabling for the development of modern biotechnology, which will be the mainstay of the center that we are launching today. Number two is to assure you that our regulatory framework, which is led by ourselves and our team of other scientists in my area. I studied the gene silencing technology, so I understand the techniques that are being used, is based on science. So we don't want stories. We don't want activism. That you eat this, you will grow beards as a lady, and men will grow something we won't mention here. We want science-based regulatory system, evidence-based, and it is done on a case-by-case basis. So I get disturbed when people say, is GMO safe? Now, what is GMO? GMO is a very specific crop or plant or something that has been modified with a very specific. So when you say they are safe, they are not safe, it is pedestrian. And therefore, we do it on a case-by-case basis. That is why the gypsophila was said no, not because of safety, but because of socio-economics. Key stakeholders, and you're becoming key ones now as an institution, are engaged in a regular review of advances in emerging in the few emerging technologies in this field of modern biotechnology for update and alignment. Science is changing even as we speak. New methodologies are coming in. New techniques are being discovered. So we engage a pool of scientists, a pool of experts that I have mentioned to continuously assess emerging issues, emerging technologies in this field for purposes of update and alignment. And finally, this center is a most welcome capacity enhancement for biotechnology development. So if I go back to my objectives as a good teacher, I want to see whether I met my objectives. Number one, to make a personal commitment. I already made that personally to the Vice Chancellor when we were having a chat at tea. So that objective has been met. Number two, to come back to my alma mater, I have met very, very important people that in my life who are my teachers and my classmates. That objective, again, is met. To make this presentation and to leave you with that message that this center is a very welcome development for capacity building in modern biotechnology. Asanteni sana. There's some email contact, yes, that.
Thank you, Dr. Ari, for the good presentation. Okay. So then we get one of the mics to him. I'm sure your presentation might receive some questions or comments. I think they are also online. So let us start with the online questions.
We have one question from Willie Moore, and he is asking, in the view of ease of doing business or natural urgency, does the turnaround time of 30 days to approve apply to all products, medical, agricultural, industrial, or even environmental?
You got it? Okay. Another question? There is one here.
Thank you, Dr. Ari, CEO, for that insightful presentation. I have a few questions for you. One is on the beta. That is the current administration. They are agitating for commercial crop, one of them being cotton. And technology they want to advance is the BT cotton. They have already gone ahead and selected counties that are supposed to do this. So what is your position? Have you okayed it? Then, my second question is on the review of the regulations. How often do you review and revise these regulations so that you catch up with the science? So maybe you comment on that. Then lastly, you said there's aspect of testing the gene if it is transferred. So probably you service for this, or you contract this service. So who are the service providers and who are the designated lab that does this?
One more question.
Thank you, Dr. Mugira. I'm George Ogendi, and thank you for that wonderful and insightful presentation. Just like most of these proposed projects that is subjected or that are subjected to EI, Environmental Impact Assessment, the information that is supposed to be shared with the public, I have seen is also being proposed that we publish a notice on 3 widely read newspapers in the country. And when you look at the approach that NEMA uses to get this information about a proposed project to the stakeholders, you will find that most of the people that are affected or impacted by a proposed project is they are not going to be able to be reading these newspapers. They are going to be listening to TV or watching TV. They are going to be listening to radio and all that. Now, in your assessment, and probably when we are looking at the proposed strategy in terms of sharing the information about GMO, about this genetic biotechnology transfer of genetic material from one plant to another, why can't we consider these other avenues of sharing information with the general public? Could it be better using barazas? Could it be better using church grounds and all that, such that the community is widely sensitized? Because these politicians are always out there to focus on the negatives, and we lose the impact of science that could have been generated through these technological advancements. Thank you.
Maybe one final question.
Okay, thank you, Dr. for your presentation. Indeed, GMOs are a solution to mankind currently because of the many problems we face. For example, the golden rice that actually has that iron in countries where they lack soil that has iron. Now, my question is, when it comes to this GMO as a regulator, what are your measures when it comes to issues of biodiversity? As a country, we don't want to lose our biodiversity because we have something called pollination. Plants, we have insects which pollinate other plants. Now we have GMO plants, and for example, we can have the GMO maize and maize which are not GMO. Now, what measures does the regulator take in place so that we don't lose our biodiversity or the original biodiversity, courtesy of the exchange of the pollen between GMO plants and plants which are not modified? And then secondly, as a regulator, for the BT maize, it produces a protein that actually causes the armyworms not to take up the plant. Now, as a regulator, and this protein has been known to actually cause allergic reactions in human beings. So as a regulator, do you base your decisions based on such studies whereby you go further and check the allergen in the recipients of these plants or of this food that has been modified? Thank you.
Thank you. I think, Dr. you can respond.
Thank you. Thank you very much, Facilitator, and thank you for these questions. It tells the interest that this is generating. I saw like Professor Song has also a question, but that will come later. Now, in view of the ease of doing business, the 30 days for genome-edited product applies across board. So it is not only for plants, it's not only for agriculture. If something comes to us that is of health, we take it through the same process. Remember, when we did the Act, the Biosafety Act, that time I think we were not very comfortable with the technology and there was quite a lot of activism. They put in a requirement that we cannot make a decision on a confined field trial within less than 90 days. And for environmental release, I think within less than 150 days. I was asking myself, why not less than? Supposing we are quick to review with our reviewers, and we have said this one can go, so we just sit there and wait for that days to happen, 150 days to happen. So I think there was a lot of fear, a lot of apprehension on the technology, and therefore that delayed that. But yes, whether it is medical, whether it is environmental, industrial, agricultural, enhancing nutrition, whatever it is, our assessment takes 30 days for the early consultation on genome editing. Beta, bottom up, yes, indeed, we are aligning our thoughts, and I think this is the wisdom the president used to lift the ban. Now, BT cotton, have we okayed it? The answer is yes. In fact, BT cotton was approved during the ban. We had even to go to cabinet, and the cabinet took a decision on BT cotton on the basis of its role in textile and cotton value chain, which is considered to be a major employer. Some of our factories, like the one that is in Eldoret, managed by Moore University, can barely cope with the need for cotton. We are importing cotton from India, from China. Now, because our cotton was decimated by this particular pest, the place we are importing from, they are growing GM. So we made a case, and that was okayed, not only by NBA in terms of safety, or NEMA in terms of environmental safety, or KEFIS in terms of plant health safety, but also by the cabinet on aspects of the role that this will play in revitalizing the cotton apparel value chain. Review of regulations, how often? Like I explained earlier, the last 10 years, these things have been in the freezer. So we haven't touched them, which is unfortunate, because I said again, science changes, and perceptions also change, and socioeconomic situations change. So we need to go back to those regulations. I have put it in my performance contract for this financial year to review the labeling regulation, because it is one that can cause a lot of uncertainties, because when we label, when we say this is GMO, what are we telling the consumer? Is it better than the other one or worse than the other one? Is it presenting a risk? You know, there are all those things that need to be considered as we go forward. But we haven't reviewed the regulations, Prof. We look forward to initiate a process of review when we start applying them. And you know, you're able to review guidelines and regulations when you apply, because after applying, that is when you realize here there is a problem, here there is a challenge that you might need to address. Otherwise, if we did them and put them in the freezer, particularly the environmental release regulations and the labeling, we need to go back to those and regulate. Testing, if the gene has been transferred, who does the test for us? We do not test and we do not commission tests. We rely on the dossier that has been provided by the applicant. And if there is information that requires to be clarified, we write back to the applicant. So we take the protocols that have been presented by the applicant. In this case, for example, if it is Egerton University in partnership with Njoro Seed Company, for example, you have given us a dossier that we have made this maize line, which is resistant to this particular disease, and we did it this way. protocol 1, protocol 2. So my team, my technical team, will look and see whether there is any place where the protocol is allowing a foreign gene to continue to the next level. And if there are questions, we do back and forth. Before we could approve the MLN maize, we had a lot of correspondence with the applicants and their scientists. So they would explain. We would even ask for some raw data. You know, those things called laminar flow things, and then my team will check. If there is something my team is not very competent with, we call upon the team of Dr. Nduko to help us to check. But we do it within 30 days. So it's a very short time, but it's a very straightforward kind of assessment that they do. Those who are in that technology can confirm. Subject to EIA, yes, Dr. Ogedi, this is an area that I am very uncomfortable with. The conduct of EIA for GMO projects, because we have already checked the environmental safety assessment, which I will describe because there is a question that has come through. Why is NEMA still doing an EIA? They have justified, they did a regulation, we respect the law, it is their role. But then you tell me, one of the questions that NEMA asks is if you are growing GM maize. Remember, their questions for EIA are very standard. Will it cause affluent? Affluent ni ile maji chafu inatoka kwa factory. You know, they are using the guidelines for doing an EIA for a factory or a hospital or a school or a building to do EIA for growing maize. Now, then they ask, will it cause dustiness? Will it cause noise, you know, or particulates in nature? So really, and you see, it becomes very relevant, but again, that is another space, so they do it. But I want to go straight to your question, can we use other avenues? And the answer is yes, because somebody can ask, how adequate is it when you throw it in the newspapers? Tell me how many people respond to those NEMA things. I normally see them on the papers. Very small font, very badly written kind of thing, a long one. You're supposed to read and depute a tick, and you know, you get even tired before you read it, unless you're really affected. That is when I see people gathering. And I want to agree with you that the way to go is to use those barazas, those kind of town hall arrangements. We went to the level of using social media so that somebody can even throw in a WhatsApp question or a WhatsApp objection, whatever kind of communication, and we also pick on that. So the answer is yes, and we want to explore that. GM Tech as a solution, what measures to protect biodiversity? When the GM crop, BT cotton, BT maize, for example, has been released from our hands, we are saying it is safe for human health, safe for animal health, and safe for the environment, because we have assessed and checked the potential of it crossing with wild relatives in the environment. It's a detailed kind of check, but yes, indeed, we check that, particularly at seed growing level, there must be distances to maintain purity. But when we release, the next levels is what is called technology stewardship, which makes sure from the developers, they are able to ensure that their product sits well with other similar products in the market. I may not be able to go to the details of that, but we also assure that. BT protein-produced toxin, this is indeed not true, or allergy. One of the checks that we do, and I want to describe this very, very briefly, we carry out, or rather, the dossier that comes, and again, an expert is here, can speak a little more about that. The dossier, when they perform feeding experiments, one of the areas that we check is they conduct what is called substantial equivalent. So a complete biochemical analysis of the GM maize, for example, BT maize, and a complete biochemical composition of the non-GM counterpart is conducted, and then they are compared to see whether there is substantial equivalence. The only difference, for example, for the BT maize, is that the BT maize is producing an additional protein, which came from the bacteria, which is killing the pest. Now, That protein is sequenced. You know, proteins are made of amino acids in a line, and the sequencing determines their character. All proteins that cause allergy, their sequences are stored in a data bank. So we query and see, is our protein resembling any known allergen? Then if the answer is no, we put a tick. Is our protein resembling any known toxin? Those proteins that cause cancer, those that cause teratogenesis, that cause loss of fetus, and so on. So we go down the checklist and putting a tick. Then finally, we also check to see whether this maize has a potential to become a super weed. You have had foreign weeds that come. There is one called madenge, which grows in some places in this country. There is the fear also from environmentalists that this may become a super weed that will not be overcome by any method of control. We also check that and also put a tick. And finally, we check whether this protein, even at its high concentration, does it have effect on non-target organism? Remember, it only targets the Lepidopteran larvae. Only the larvae. It doesn't affect even the adult, because the gut of the larvae has specific receptors for the toxin, for the protein. So I think science is so specific. You know, science is not random. Science is not kienyeji. Is not kuzunguka ile. It is very specific. It only targets that. So tests are therefore carried out. The protein is fed to bees, to earthworms, to birds, to mammals, to fish. Their anatomy is checked, their liver is analyzed, and check whether there is any effect. When that effect on non-target organisms is also, we put a tick. Now we can package this and say this can go to the environment for cultivation as a crop, can come to the dinner table as a meal. Thank you.
Thank you, Dr. for the elaborate responses. And I think, as you have rightly said, we do that risk assessment to check whether it is allergenic, toxicity, all those procedures are there, and they are science-based. Now, maybe I may ask if there is an online question from our online audience. Okay, go ahead.
There are two more questions from the online audience. One, would you specify or clarify on the concerns that have been raised on BT cotton production in Burkina Faso? Then the other question is from Mandiziza, is saying, seeing the issue of GMOs affect the public, is there any consumer awareness on the use of GMOs and the implication on human health? If the awareness campaign has been conducted, what was the overall reaction from the public?
Okay, over to you, Dr.
Thank you once again very much for our participants online. Would you specify concerns of BT cotton in Burkina Faso? When a technology is developed, the protein that, or the cassette of genes, that gene that makes the protein that kills the pest, it is put in a variety. So there is a hybrid variety by company X, company Y, where this technology is put. So in the case of Burkina Faso, what went wrong was the breeding of the variety onto which the technology was put. It was put in an inferior variety that was producing shorter length of cotton. So it has nothing to do with the BT technology. It was a selection issue of the background. Remember, even if you feed your cow with very high level feed, if the genetic is not correct, Professor Bebe will tell you, you will not get high yields of milk. You have your Congoni, the local Zebu cow, which genetically has the capacity of producing 2 liters. Even if you brought whatever feed, it cannot go beyond that because the genetic background is weak. So that was the case in Burkina Faso, and it was a mix-up really that happened inadvertently, and that is why Burkina Faso discontinued the cultivation of BT cotton. It must be understood that it was not on the basis of the technology. When I say that, that is when people start telling me you are now a promoter. I'm still a regulator, my good people. Yes. That's right. The gene that is responsible for fiber development has nothing to do with the BT gene. The next question, issue of GMO affecting the public. This is the area that I want to admit and agree we have not done well, either as an institution or as a community of practice. We have not engaged the public because a lot of times, and he has asked, the person who asked this question also asked what has been our experiences. We went to the devolution conference 2 years ago when it was in Kirinyaga, and we randomly went around with a mic and we asking somebody, what is your view about GMOs? And the person would say, those are the things that are coming to kill us. They are causing cancer, and you can hear really when the assessment is done, it doesn't. Then we ask them, what is a GMO? And you put the mic, and you hear all manner of answers. So even those who don't know what it is, what they know is that it causes cancer, it kills, which is very negative. And this is an area that I will say again, Kenyan experience, this is what I can speak about, and it is reflected across the globe. We have not done very well. Because for us, for example, in Kenya, the last 10 years, like I said, we have been in the freezer. We didn't have anything to talk about. Now we can talk about BT cotton. We can take even people to the field and tell them, this is the BT cotton, this is the non-BT cotton, look at it and make a judgment out of it yourself. When we didn't have anything, we used to take members of parliament to South Africa, and as we drive through the maize fields, members would ask me, where is that GMO? Then I tell them, all those maize that you have seen, even the farmer who is hosting us, the ugali that he has made is GM maize. And then members of parliament would tell me, we have not seen. Atujaona iko happy. I think they expected to see something like an ogre. You know that thing that we studied in primary school, a huge human being with 2 mouths and 1 eye. What the people of Kimurto would call Chemo seat. So that is the perception. I want to agree with the online participant that this area needs quite more work. It is a very expensive engagement because you must have the meetings and discussions, the town halls that Ogedi has proposed. And we want to go that direction. Many times when we make presentations, we were with Professor Mulwa meeting the Agriculture Committee of Parliament. After we made our presentations for a whole week, they came and asked us, so that is what it is all about. I don't know what they knew before. And they even committed to increase my budget. The NBA is the only agency in government that has received an additional budgetary allocation this financial year in the State Department for Crops because of the need for public education and awareness. We haven't done well. We're doing something about it. And we want to recruit a large number of scholars like yourselves who can speak the science and allay fears that are not science-based so that people can make decisions on the basis of knowledge. The other reason that we haven't done very well is we also come from institutions and also as scientists. We have been blamed as scientists for not telling it. But you see, scientists always back their saying with facts and evidence. So you cannot stand there and start saying people grow horns because that is not science. So the science must also analyze your data, and therefore we want to learn, and we shall be recruiting you. One of the key areas, and I appreciate the center that we are launching today, is the technology transfer, which will also go with community engagement. Thank you.
Thank you, Dr. Ari. And I think on the area of doing capacity building of the Kenyans, we are lagging behind. So we need to be even on the KOT. Yeah, but the other day I saw Dr. Joyro Jiang and Professor Duor on KBC. I think the scientists are doing their part. So at this point, let us appreciate all the presenters in the morning session. So I want to hand over the mic to the MC, Dr. Mafura.
Okay, thank you, Dr. Nduko, and thank you, Dr. Mugire. That one is a very sensitive topic, and even if we just allow it, it can go up to evening. I think right now we wish to break for our lunch. We need to go break for 1 hour. So if possible, let's be back by 2:30, so that we can have to catch up with the other sessions. For the online viewers, please make sure that you join us at 2:30, so that we'll be able to continue with the workshop. Thank you so much. So let's break for lunch.
We want you to be there. Practical training that relates and fits so well in the current world.
Egerton, teamwork!
Egerton University, transforming lives through quality education.
I do remember Egerton University. Those memories fill me with gratitude. Egerton University placed me right inside a pool of professionals, gave me lawyers, doctors, business partners, everyone I need to continue advancing on speed dial.
I'm proud to be associated with this institution, Egerton, and it is shining, and anywhere I go, I normally meet people and they tell me they are from Egerton. Great people who have succeeded in different areas.
Success is like waking up to wash your face in the morning, only if you go through Egerton University.
We welcome those joining Egerton University. And for those not yet, what are you waiting for? Go to COOP's website and choose Egerton University. There is a whole world waiting to hear from you. Let's give you a voice. Egerton University, transforming lives through quality education.
Egerton University houses the following faculties: Agriculture, Arts and Social Science, Commerce, Education and Community Studies, Engineering and Technology, Environment and Resource Development, Health Sciences, Science, Faculty of Law, Faculty of Veterinary Medicine and Surgery, Institute of Women, Gender and Development Studies, College of Open and Distance Learning. Now, under the Faculty of Agriculture, there are the following diplomas: the following are the degree programs: master's programs, and doctorate degrees. Under the Faculty of Arts and Social Sciences, we have diploma programs, degree programs, master degree programs, and doctorate programs. The Faculty of Commerce has the following programs: diploma programs, degree programs, master's programs, doctorate programs. The Faculty of Education programs has the following: diploma programs, degree programs, master's programs, doctorate degrees. The Faculty of Engineering and Technology has the following: under the Faculty of Environment and Resource Development, diploma programs are bachelor degrees, master's degrees, doctorate degrees. Under the Faculty of Health Sciences and Programs, we have bachelor degrees, master's degrees, and doctorate degrees. Under the Faculty of Science, we have diploma degrees, bachelor degrees, master's programs, doctorate programs. Under the Faculty of Veterinary Medicines and Surgery programs, we have academic programs, upcoming undergraduate and graduate programs, short courses. Under the Institute of Women, Gender and Development Studies, we have the following: last but not least, we have the College of Open and Distance Learning Programs with the following: Faculty of Law with the following courses: Egerton University, transforming lives through quality education.
We want you to be there. Practical training that relates and fits so well in the current world.
Egerton, teamwork!
Egerton University, transforming lives through quality education.
I do remember Egerton University. Those memories fill me with gratitude. Egerton University placed me right inside a pool of professionals, gave me lawyers, doctors, business partners, everyone I need to continue advancing on speed dial.
I'm proud to be associated with this institution, Egerton, and it is shining, and anywhere I go, I normally meet people and they tell me they are from Egerton. Great people who have succeeded in different areas.
Success is like waking up to wash your face in the morning, only if you go through Egerton University.
We welcome those joining Egerton University. And for those not yet, what are you waiting for? Go to KUPP's website and choose Egerton University. There is a whole world waiting to hear from you. Let's give you a voice. Egerton University, transforming lives through quality education.
Egerton University houses the following faculties.
Science, Dr. Steve Odor. So Dr. Steve Odor, please come so that you can be able to chair the session. Welcome.
Good afternoon, dear colleagues, and welcome to our third session of our today's workshop. I'm privileged to have been given an opportunity to facilitate this workshop, third session, which focuses on a joint research funding proposal development, and to present to us their observations on this very important area. We have two presenters, Professor Patrick Okori, who is the Executive Secretary of RUFORUM, and the second presenter will be Professor Zhu Wang from Nanjing Agricultural University in China. It's my hope that Professor Okori is already available and is with us virtually. Professor Okori, can you get us?
Yes. Yes, I can hear you.
Thank you so much, Professor. Welcome to this session, and just to give you a brief of who Professor Patrick Okori is, I wish to state the following. Professor Okori is the Executive Secretary of RUFORUM, and he is an accomplished Ugandan scientist known for his leadership in agricultural education, research, and innovation. He holds a PhD from the Swedish University of Agricultural Science and he has excelled as an educator and a researcher at Makerere University and as a principal scientist at Egerton. His expertise spans crop improvement, technology, scaling out, and agricultural systems across Africa, which contributes to the development and commercialization of numerous legume varieties. Professor Corey's visionary leadership has left a profound impact on various institutions and initiatives across the continent. So we are highly privileged to have him with us to give us a talk on this very important area on overview of research funding opportunities. So Professor Corey, you are most welcome to give us your presentation. Thank you.
Thank you very much. Let me try to beam what I appreciate. Thank you, Dean, for the opportunity to share with you. Let me see if I can screen. It's giving me a hard time. Just a minute. Sure. Can you start the next presenter? I'll come in after him. I'm having a system hiccup so that I don't delay you. Thank you. Chair? Yes. May I request you just so that I can change my computer? This one is not behaving well. Can you shift to the next presenter? I'll come after the presenter. Thank you.
Okay, I'm not very sure whether he's on already.
If he's not, just give me 2 minutes. I'll be back with you, sir.
I think we can give you 2 minutes so that we don't disturb the flow of information, please. we can hear you, Prof. That's fine. Then we can just go ahead. Most welcome.
Okay. Thank you. I had to change computers. Some of these toys don't behave all the time. I apologize. Really sorry about that.
You're welcome. No problem, sir.
Thank you. Okay. Can you see my screen?
Yes, it is coming up. Yes, we can see it.
Okay, let me try to put it in presentation mode. Is it clear now? Yes.
Yes, it is clear.
All right. Good afternoon, and I would like to, my name is Patrick Okori. I would like to thank the Vice Chancellor of Egerton University, the management and staff for inviting me to be here. Professor Kibwaje, thank you so much. Professor Nancy Muigai, thank you. And everyone else who is running the master ceremony, I recognize all participants here today. I'd like to talk to you for a very short time, sharing with you some thoughts on really funding opportunities for agriculture and higher education in general. Before you do that, I think it is important whenever you're raising money to think about what it is all about. The main problem is not that there is no money. The main problem is generally that you present your case. Money is only available for people with causes, not for causes with people. And so you must match causes and people. But many times we present the people without the causes or the causes without the people. That's why it's important to have this context. Right. Now, in the case of Africa, agriculture remains a very big area for investment. Up to at least 2040, almost all our national investment documents, including Kenya, talk about that. Our population remains young, less than 60%, less than 35 years old, and more than 70% of our population is generally less than 40 years old. And so we need to focus on how to create jobs for these people, wealth opportunities, and a more sustainable earth. And that means agriculture is key to this whole business. Health and nutrition is key to that business. Financing and wealth creation, job opportunities, these are the top agenda for the next 2 decades or thereabout. I repeat them. How can we feed these people? How can we create jobs for these people? How can we ensure that there remains a thriving economy or earth to look after them? Because of that, we need to think about having renewable wealth production systems. These will be needed in all kinds of forms. We need to feed and grow the continent, and as I've said, we must create the jobs. Now, if you come to think about these investments, I'm still focusing on having your context right. How do you pitch your message? There are known investment opportunities for transformation. Most of the times, there's really nothing new. It's pretty known, the things I have talked about. On your left side of the screen, or right, depending on where you are, you can see East Africa there. We have still quite a bit of land that we can expand to grow our agriculture. And we are actually using the shorter bar. The shorter part of it is how much we are using, and the greener part is how much potential there is. We're actually doing less than 50% of the potential that our land can do for us as a people. And if you look on your right, these figures are from 2006, but they're still correct up to now. The only way out of moving people, millions out of poverty, is really, among others, to grow agriculture. Land expansion remains a key thing, and that's what we have been doing for decades. But we must focus more on improving the yield. So in summary, I wanted just to send these 2 messages, that the full potential for our Africa's arable land, and Kenya also in particular, is well more than 100% for most of our countries. This is where technology can play a big role. But there are economic challenges that we must think about, because to grow agriculture and address health, nutrition, income, jobs, all these wonderful words, you must move away from conventional ways of doing things into non-conventional. And this is where we need to rethink how we do certain things. And Kenya is leading one of the ways in that, using digital technologies, which are seen as breakthrough technologies that open equal opportunities for everyone. In the case of agriculture, it still remains inadequate access and or inappropriate use of even the conventional inputs, such as land, labor, seed, fertilizers. All these things are still being utilized at a very low capacity. This is just a story from Sustainability, a paper published in 2022. It's a measure of how effective our economy is growing. It's called total factor productivity, a measure of how you use your inputs to produce outputs. I want you to focus on the column in red. Now, the numbers after 1 there tell you the extent to which you are growing actually. What you will see that for the rest of Africa, we are all growing, especially East Africa. We have done quite well when it comes to technology progress, and most of this is due to digital technologies. But when you compare to the rest of Africa, we've grown by about 13%. The rest of Africa, we are doing about 8, 5, 6. So we are doing quite well on digital technologies as such. But I want you to look at the first column and how much our agriculture sector is growing as a whole. You notice in most cases it is growing at a maximum of 4%. North Africa is doing 8%, Central Africa almost 7%. Our region, including Kenya, we're just doing 4% or 5% at most. Now, this is annually, and that is not going to help us to solve the development challenge, because Africa must grow at least 10% if it is going for 10 years, if it's going to move people out of poverty. So we are less than that. And that means our story must change and how we do things. In fact, whereas the sector promises to be a $1 trillion industry by 2030, it remains a hope. Why is that so? Because of the kind of trends that you can see. When you look at the extent to which technology efficiency, how much is human knowledge and the capacity of the people translating science into development, you see that we are making some progress, but in many cases, very limited progress. When it comes to access to technology, we are even worse off. So very few people are using improved technologies, including in East Africa. We trail in many parts of that data. So the main message here on that slide on the right is that when you project up to 2030, it is seen that Africa's growth in general, the GDP growth, will exceed that of the EU, estimated at about 5.9%. But most of this is focused on large industrial investments rather than roads and things like that, rather than on agriculture per se. And so poverty will still remain an issue even when that is true. So with that kind of context, you now know at least that you need to think very broadly, what is the development challenge? What must I think about? What must our institution be known for? I'm arguing these days among our foreign member universities to think of some 2, 3, 4 things that when you close your eyes, you are known for, whether it's Egerton or Makerere or wherever. If you close your eyes and see the buildings, we may not be seeing what needs to be done. Egerton, for example, is very well known for certain things. When you close your eyes, you see it outside there. A good example is what Professor Mwingai and others are doing. You're rolling out alternative methods of stabilizing the food security system of Kenya. That's a good thing. That and others, Professor Kibe and his team and many others I know, they are all doing things that are related to stabilizing food security. And so you seem to be moving on your needle towards alternative systems for delivering agricultural technologies and development thereof to the people of Kenya. If that's your thrust you want to go on, put your energies around that. And my argument is rope in health. Don't forget about health, because health and food and nutrition and agriculture are the same. I just said that closing the funding gap, there'll always be money challenges. And this is just a slide to share with you. This is a picture I took someplace in Zambia where we were trying to release groundnut varieties, and these farmers were excited about them. But anyway, you can see that investing in research is close to 51% able to change the way the sector is growing. But you can see that investment in other sectors where government puts a lot of money, for example, AIDS will only change the sector by 2%. Reducing conflicts where governments are running all over 18%. Trade that everybody talks about is just 20%. So if agriculture is not, research is not invested in, and universities are not part of that process, this story will continue on beyond our lifetime. Now, just a minute. Why isn't it behaving? So, my computer is stuck. So as we continue, I'm having a technological hitch here. There are a number of questions that you might want to ask. Why then should you focus on agriculture, and where should you be focusing on agriculture? My computer is stuck. I don't know. Can you hear me? Yes. Yes. Yeah, I'll just talk without the computer because it's not behaving. So if you want to ask this question of what you should be doing, then in terms of, I'll share my slides that can be shared later on. One needs to ask, where is the money going and what is it doing? In the case of Kenya, a lot of money coming into the agriculture sector or the economy as such, the agriculture sector. Kenya, Rwanda, Tanzania, they receive about 89% of the money which comes from philanthropy. These are agencies that are supporting these economies, compared to the rest of the East African countries that receive only 11%. So Kenya sits in a very attractive position. When you look at money being spent by private sector, this is another group that we must rope in to fund us. Again, most of the money has been growing by different proportions, but most of this money is actually not in agriculture. For example, in Kenya, about 10% of the money that has been coming between 2013 to 2019 or thereabout has gone to real estate. The largest part of it has gone to consumer spending, and about 15% into technologies, but none of it has gone into agriculture per se. And so, whereas private sector is there, the funding is there, foreign direct investment is coming in, none of it is going into agri-research as such. And so it is important that we spend money and time on things that will attract people to spend. I wanted just to air a little bit and share with you one slide. I'll share with Professor Muigai to share with you people. And it basically shows that whereas weather is a threat, actually, maize, which is a staple food in most of East and Southern Africa, will benefit a little from climate change if we remain at 1.5. But if we go at 2 degrees, we are going to lose roughly about 10 to 15%.
A disconnection or something? We are not hearing.
Now, resources, money. You need money basically to underpin and
the disconnection challenge, network challenge. I think there's a challenge with the connection network. Yeah. Okay. Prof, we can't hear you. We can see the slides changing, but we can't hear you, Prof. But he's talking.
No, he's talking.
No, I think it's from his side.
You can text. Paul, can you text?
Maybe meanwhile. Maybe we may have to request Professor Wang, if she's on virtually, and we may connect with her so that she makes her presentation, and maybe later on we may go back to. Oh yeah, I hope we'll be back. And we
Recording stopped.
Prof. Okori seems to be back, eh?
I was just about to finish.
Okay. Welcome back, Prof.
And you can see my slides?
Yes, we can see them.
Okay, so I'll try to rush through about 3 more slides and I'll be done. So I was just showing you this, that Kenya is leading in terms of philanthropy, and you can see where most of the money is going. None of it is in agriculture. And I just wanted to raise the issue here that when you compare Africa and the rest of the developing world, we are still spending so much money on expanding land rather than on technology. And this meeting is about technology. When you compare the rest of the world, they spend more time on technology rather than land. And that's why they're able to feed their people. And for Kenya, this is what I was saying. This is Kenya here. You can see on a 2-degree rise, you're going to have just about 14% loss in yield of maize, compared to if we never had a climate change scenario. And so we have a wind that is coming against us, bad weather, and we must prepare for them. So for Kenya, you must tie food security, nutrition security, income, and as I've said again, health must come in. So the units of agriculture, the units of human nutrition, and the units of agriculture, and of natural resources, they need to begin speaking together if we are going to mobilize the kind of monies we need. The time for unit-based types of research is gone. We need to be thinking transnational types of research. And then finally, like I said, universities are receiving little money than they used to. There are less money for units. There is more money for larger groups. And for that means that universities need to put in place an institutional resource mobilization, which is focusing on 3 things. You must have a brand you are selling. What are you known for? Number 2, there must be an office or organizational development, the kind of thing that Prof. Nancy is doing here. And then communication and prospecting on a regular basis. Somebody has to be providing the Vice Chancellor's office with this data to the academic registrar. Ideally, on a weekly basis, there must be somebody monitoring that. This is how international research survives. This is how it works. And that cartoon on the right is really the kind of things that have come out of the, that you need to think about when you're resourcing. So as I wind up, to get it right, it's important, like I said, you must know your customer. The context must be okay. You must, and your customer in this case is the end result. Who is your ultimate beneficiary? Know them and their needs. 2, think about who the donor is. They also have all kinds of scope. And this is why you need a development office, which helps you to prospect and know well in advance. I have been in international research, and the only reason they get money more than us most of the time is because sometimes they know about these calls even 2-3 months before they are out, and they have been preparing. And by the time you're going after those calls, you are already too late to compete. And so if you don't start, if you want big money, you hunt differently. I always say if you're hunting buffalo, you don't hunt like you're going after antelopes. The strategies are different. Have your priorities and the priorities of your partners nicking. What capacities do you have? Share that and let it be well placed. I liked your video talking about your university and the kind of things you have. Those 1-minute videos are so important. And then, what capacities do you not have? Be honest about that. And then teams are preferred rather than individuals because you are going to cost leverage. You are going to provide opportunity for scaling and, more importantly, opportunity for learning. In terms of funding resources, there are broadly 3. The overseas development agencies, these tend to be very large grants, $30–40 million type of things, sometimes 100 even. But they require teams, they require multi-partners, they are cross-regional, they are very complex and highly competitive. It is highly unlikely you can win this as an institution. You have to be part of something.
Recording in progress.
2, government. This tends to be small to medium and short term, but they are mostly for last mile delivery. Like I always say, they will fund you for some little few things. You spend so much money on them, but the return is less. However, you get the political capital. So you need to focus on them too. And they're good for young starting up scientists. Then thirdly is the philanthropy. These guys have big money sometimes, and most of the times. But you've got to engage them. This is where having a strategic marketing office is very important. They need to be on your mailing list, receiving your emails, receiving letters from the top management of the university, and so on. They need to know you. Money is given to people who are trusted because it is an element of trust. And so building this helps you a lot. In terms of implementation, Egerton, you're already doing part of this. You're already doing some of the things I was talking about, institutional resource scouting and having a management office. You can call it whatever you want, but you need somebody who does that as their go-to job. You must have strategic partnership and marketing function established. We usually call this communication, but I think these days it's called slightly different. And I've just given you one of the ways it is called. You have to build this both locally and overseas. And this is where your alumni network plays a crucial role, like I had on your video. And then you must downscale this really to the units that are going to implement all of this. Finally, as you write these winning proposals, please remember you're going to fail several times, but don't give up. You have to keep going. I have failed several times, and I keep going back to those failed proposals. Using the corrections I was given, I improve them, and eventually I get the money. So large teams will help you. Agriculture, your concept notes, start from ideation and grow your case. Reach out and to others to review. Don't be afraid of negative comments. It's part of the growth deal. Develop your full proposal. Take your time to develop it. Last minute submissions are never useful. They never win anyway. You'll be lucky to get through. But most importantly, you need to message these days using conceptual diagrams. You need to have a results-focused programming. People must see your outcomes and outputs. It's no longer useful to write a proposal where your output is the number of students graduated and the number of papers you've published. That is good, but those are elements to the ultimate. That's why you must show that pathway to delivery. And many of our universities need to develop a very strong monitoring and evaluation unit that tells your story. And I can see the Vice Chancellor and his team have put some of these in place. And this is necessary for large grants. So, ladies and gentlemen, I think that's what I wanted to say, just by ending by saying that please remember, if you want large grants and large money, you need to be part of a partnership, because less than 1% of Africa's GDP is actually spent on agriculture, on investment in research, basic research especially. And so if you want to get more money, you must be part of another circle. Two, we need to build capacity of our young people and get them on board, because we have very few researchers per million compared to the rest of the world. That means you will never have capacity to do everything. I've given you a few people you can start up with. I think that as an institution, it's important to map out who your key partners have been for the last 10 years and identify a focal person for that. I've given you the one CGIAR for example. And this is one place you can go to. They like partnerships and they need universities to work with. And then you have overseas development assistance. Generally, these guys like to talk to the Vice Chancellor's office. These are the big money guys, the USAID, Japanese aid, and all that type of things. They prefer to go through the Vice Chancellor's office. So having your international program directly linked to this type of thing is very useful. Then you have private foundations. These are really don't care too much. You can go through the partnership office or you can go through the VC's office. But good examples that would like to support the kinds of things you are doing include Bill and Melinda Gates, the Rockefeller Foundation. MasterCard is right now one of the large ones around. The Syngenta Foundation. I could go on and on and on. But this list, like I said, keep on changing. You need a strategic management office which helps you to monitor on a regular basis. Finally, this is my last slide. I remember the words of Franklin D. Roosevelt, who once said, it is always important when you're thinking about something to keep your eyes on the stars, but keep your feet on the ground. In other words, think big, but be real. And I'm sure you'll be able to get the funding. Thank you very much for your attention.
Thank you very much, Professor Corey, for that wonderful talk, in spite of those challenges from the network connectivity. But I think the talk was really nice, and it has really benefited us so much. I think the keyword statement I wished maybe to reiterate is that whenever you are applying for funding, there will be lots of times that you'll fail, but never give up. Keep on trying, eventually you'll succeed. With that, I want to open the floor for some comments and questions, both virtually as well as from within the room. You all satisfied? Yes? Could you please give your name and then...
Thank you very much. My name is Bernard Olo of Dairy and Food Science, Egerton University. I'd like Prof to share some comments about data shared on where investment would bring most value for money, and which particularly agriculture. Whether there is or there are fora where this information is shared, especially political wing, to persuade them to make investment in agriculture and transform lives, and yet it's possible huge investments in research and investments in a aura this is happening, to continue to persuade the political to ensure this investment happens. Thank you.
Thank you for that question.
Chair, may I request you to repeat the question for me? I lost most of it.
Okay, then come over.
Hello, Professor, can you hear me?
Yes, please, go ahead.
Thank you. My name is Bernard Olo from the Department of Dairy and Food Science here at Egerton University. My question pertains to the statistic you showed with regard to where investment would have the most value for money, and that was in the area of research and agriculture. And my question was to ask whether there is a fora that is dedicated to persuade, particularly the political wing, to ensure that we encourage them to make sure that there's more investment that is happening in these two fronts, because by the end of the day, these two fronts would make the best investment decision for the development of the masses, which is their main aim as the political wing. Thank you.
Thank you.
Thank you. Any other question? Then maybe, Prof, you can respond to that.
Thank you very much, Bernard, for your question. Actually, there, let me call it 4 levels at which you can influence policy. The first level is this one you're in. It is important that your head of institution understands what you're doing and can sell your vision. And because as the CEO of the organization, he goes to lofty meetings that the rest of the managers may not go to. So ensure that your leaders are fully aware of what you're doing, because wherever they go, they have a very high chance of meeting somebody who wants to talk about this. And because agriculture, health, nutrition, jobs are a big thing right now in Africa, for the next decade, and agriculture playing a big role, it's important that your deans, your principals, your vice chancellors fully have this. I always say it this way. Make sure that you develop as a department, as a unit, a small half-pager that your vice chancellor can have about who you are as a unit and what you have to sell. If that's not available, it's difficult for him or her to do so, especially if they are not experts in your field. Infographics are the easiest way to do things. So these are A3 type of, these small half A4 documents. If you have that, it's going to be very helpful. So institution, they must know. 2, you must make sure that your local government also is aware. And the kind of thing that was held when you had a National Forum Day not so long ago, Egerton is leading actually in the entire Forum family, and we're grateful for the university leadership and the team at Egerton for that. When you hear people coming, if I recall, there was somebody from the office of the president or vice president, from the office of the Minister of Agriculture, when they come and they hear such things, when donors always fly by night, those are the first places they go to. They will have you to sell. And if they have these brochures of who you are and what you can do, selling your pitch is going to be a lot easier. And then finally, it's about making sure, like I said, at the national level, it's about making sure that you're doing the prospecting at the highest level. Now, your vice chancellor is also a member of RUFORUM's board. They own RUFORUM, in other words. Every year we have a meeting on financing higher education and things of that nature. And there's an opportunity to actually present these cases there. And usually we invite the ministers from the partner countries to come and participate in this type of thing. So RUFORUM can also be an advocate for you. But again, arming RUFORUM with what you do is the best. And in the case of Egerton, RUFORUM is pretty aware of what you do. So selling you is not a difficult thing. However, selling yourself to the donors even pays a lot more. But that requires, like I said, good institutional setup where you can sell if somebody says, I want to come and see. 2, it requires your leadership to across the board to be fully aware and to be engaging with different partners in that. And then the rest is about talking. There are so many types of fora. I must admit, in my life in this business, you don't know which one will yield fruit. Sometimes the one you think is least is the one that yields fruit. I just wanted to encourage that next time you have a national forum day, please invite the big guns like MasterCard Foundation. I saw you invited them last time. Invite USAID, invite Japanese aid, invite CEDA. There are so many others. Invite those overseas development agencies just to see the work you do. They might not give you anything in day one, but they have taken note. I found it as helpful to walk down the road that way. So there are various fora, but the onus is on you to do the job. Thank you.
Thank you, Professor Corey, for for that response. I think we have one online question, and our team.
Hello.
May ask. Yeah, there is one online question. Yeah.
Yes. Can I go ahead?
Yes, please.
Hello, Professor. I just have, I'm Dr. Georgia Sudi from Kenyatta University, and I just follow up with the same question. I think, I don't know if you can give a comment on this, but I'm just wondering, what do you recommend,
Recording
especially
stopped.
for the young, what we call the young upcoming researchers, especially in terms of collaboration? Sometimes it becomes so difficult to find even a collaborator willing to give you a go-ahead to start. And especially the CGR Center, sometimes if you have no idea even who you are going to meet, it becomes so difficult. So what do you encourage us to do? Thank you.
Thank you,
Recording in progress.
Dr. King Asudi. I appreciate your question. I had the same challenge when I just started up. I think, have you ever heard of International Fund for Science, IFS? If you have not, please Google them up.
Yes, I do. I actually have a funding. I actually have a research fund with them.
Okay.
Yes. Yes.
So I encourage all young researchers, fresh PhDs. When I used to be at Makerere as dean, I kind of enforced that. If you're a fresh graduate PhD, you have to apply for IFS. And I made it my business to help you. You'd fail 2-3 times, but they all got the money. The PhD students who are in the middle of or towards the end, we also enforced that they had to get an IFS grant somehow, somewhere. And we used to lead on that. And I'm sure Kenya leads one of the leading recipients of IFS grants. Why did I start with that? The nature of the IFS grant, the structure, that proposal you fill, is just about the structure you will use throughout your life. And if you can use it and fill it and complete it and get a grant, you are on your way to winning large grants. Now, that's one. Number two, as a young researcher, you would also need to focus a lot on publications. Now, many people want easy publications. They are good, but aim for the higher order. People call them higher impact factor. I don't so much believe in that. But look for something very traditional and put your paper there. The chances that once your paper goes there and you seek for partnerships, everyone wants something that is already glittering. No one wants to start from ground zero. So many of our young African researchers, they publish a lot locally. When you Google them up, many of them are in the gray literature type of places. Please enforce that, ensure that in your first 5 to 6 years of publication, you are focusing on high impact factor or high level types of journals. Those will need you to be handheld. You may not be the first author, but the fact that you're part of a group will help you a lot. It means you have teamwork. Now, the third thing you need to focus on, you're asking me, how can you go about meeting these people? Well, elders lead you everywhere in any society. And this is where I was talking about a strategic marketing office. The strategic marketing office of Egerton University can contact the strategic marketing office of NSCGIR. They'll work with you. They are looking for those partnerships. If it is institutionalized, it's believed in, it's taken more seriously. If it is on individual basis, there is no full guarantee that they will partner with you. However, as RU Forum, this is also part of our job. Our job is to help you to link up with all kinds of partners. And I want to encourage you through the office you have, the graduate training program and the program Prof. Nancy manages, among others. When there are these European Union calls, please do participate when she invites you and to pitch in for them. They usually have partners across different types of organizations. The reason we do that, many fail, but that is the starting point to build partnerships and knowledge. And finally, whatever little money you have here and there, that can allow you to go to a scientific meeting. Never be shy that they have asked you to present a poster. You don't have to talk everywhere. People just have to see the kind of information you have, and they will be willing to partner with you. Where I come from, they say good meat sells by itself. No one advertises it. So if you are doing a good job, even if you're having a poster, I have been to meetings where I presented a poster as a graduate student. It opened me up to so many other geneticists around the world because they wanted to work with this young person who wanted their help. And so if you reach out, people reach out to you. So reach out to Forum. Our partnership office is available. You can come through your national office, through the dean's office. All deans are actually points persons of Forum. That's my final message. Get to the dean will contact us, and we will try to see the opportunities that are there. I am fully aware of the CGIAR system. I can also connect you, and I have an idea of the goings on that is happening. What we want to do going on after is that when we receive information from the CGIAR intelligence, which is much more deeper than what many of our organizations have, we'll be transmitting this generally to the strategic marketing offices, and they can downscale it down to you. I think that would help all of us to go forward, and also other donors, not just CGIAR. Thank you. I hope that helps you.
Sure,
Dr.
sure,
John.
yes.
Thank you, thank you.
That was Shadrach, or I'm seeing there's somebody who has raised up his hand.
Shadrach Imai.
Yes, Shadrach, just
Yes.
go ahead.
Yes, Shadrach. I'm from Kalro, Kakamega. My question is to Prof. Maybe you can comment on this or share some insight. What do you think of this crowdfunding research? General crowdfunding research. Yes.
Thank you, Shadrach, and thank you for calling from Kakamega. Two issues. One, crowdfunding is becoming interesting. But for most of the research that we want to do, you will not be able to use it immediately because people give you $2, $100, $50, etc., etc. If you want to do meaningful work, you need definitely more work and so more people. So it can be very discouraging. My suggestion, which is something we wanted to talk about even in our upcoming Cameroon meeting, among others, with our deans and so on, is how to use our alumni to start crowdfunding to fund initiatives in our universities. So, for example, the Egerton University alumni, you can have that, and it can start raising $2 or $4. The amount of money people spend on beer in one evening, somebody can give it up. I see them doing that for newspapers, for children in Kenya, and so on and so forth. So crowdfunding is good, but the amount of money that you get is little. It needs something else for you to make it work. You need to have a proper investment vehicle that you can then use it to grow that fund as well. So it is good for sustaining, but very rarely is it going to help you to start afresh unless you are crowdfunding with large source of money at the same time. Then it will work. The second thing is that if we want to succeed with crowdfunding, it's got to be on one or two strategic areas we want to put our interest in. That way we can pool resources. So for example, if we focus on research for the drylands, it is highly likely that even other large international concerns, they will not just come to crowdfund, they will come to cost leverage to add their money to it. So if it is topical and not just the flavor of the month, but a very important thing, you will attract people to it. But pooling resources over and across national boundaries have got their own limitations, rules, and regulation. So in short, the whole management of crowdfunding at institutional level needs to be thought about. Many of our organizations are still not yet quite there in terms of policy, the frameworks, how do they manage investment vehicles. All these things need to be sorted out. And for Cal Republic organization, that's a different ballgame altogether, I think. Over to you.
Thank you so much, Professor Okori. I think that marks the end of our questions and interaction with Professor Okori. Let us give Professor one more round of clap. And
thank you so much, too.
We really appreciate Prof. Thank you very much. And with that, I would wish that we move to our seventh presenter, and that is Professor Ziu Wang of Nanjing Agricultural University. Professor, can you hear us?
Yes.
Great, Prof. Welcome.
Hi, Professor.
Thank you so much.
Can you hear me?
Yes, we can hear you.
I'm sharing my screen. Can you see?
Yes, I think we can see your screen. It is visible. Maybe you change it into the presentation mode, if possible.
The screen show. I will show my.
Yeah, and just before.
Okay, slideshow. Can you see?
Yeah, just before you start talking, I would wish to formally introduce you to the group. Professor Ziu Wang is going to be our seventh presenter. Professor Wang is a distinguished wheat breeder and geneticist with a prolific record of over 100 papers, 6 books, and more than 30 patents under her name. Her remarkable achievements in wheat breeding have earned her accolades, including awards for technological innovation and science progress. Professor Wang holds pivotal leadership role in various agricultural institutions, including as a deputy dean and director of key laboratories. She is also an accomplished editor and committee member in the field of genetics and crop evaluation. And I therefore wish to warmly welcome Professor Wang to give her presentation. Welcome, Professor.
Thank you, Professor. Thank you, Dean. Thank you for the organizing committee of the workshop. This is a great workshop. I have listened to the talks, many talks, keynote talks, from the keynote speakers, and also the talks. The last talk is about for the application of granting. So for me, it's also very helpful, very informative. And many congratulations to the Egerton University for the approval of the International Center for Genetic Engineering and Biotechnology. So I can see from the name list of the participants, many old friends. So we met maybe in 2000. I visited Kenya, visited Egerton University twice in 2009 and in 2017. And also I host the friends from Egerton University for their visit to China. So we are planning another visit to Egerton University where we'll have a group led by the president and also from the scientists or professors from the Belt and Road Joint Lab. So thanks for the invitation. So my talk will be very broad in broad areas. So I'm talking about the Kenya-China collaboration experience. So take the China-Kenya Belt and Road Joint Laboratory on crop molecular biology as an example. So my name is Ziu Wang. I'm from Agricultural College of Agriculture, Nanjing Agricultural University. I'm working on wheat genetics and breeding. So I'm so happy to participate in such a workshop. And I will talk about actually our collaboration history and our achievements about our collaboration between I think we only focus on the two universities collaboration. And I will also talk something about what we will do in the future. So I am from here. This is China. I don't know if you can see my. can you see? I don't know if you can see my. So I'm from Nanjing.
Your slide, please.
In the center center part of China, along the Yangtze River. So the Nanjing Agricultural University is a very old university. So, and we have a long history of collaboration between our university with Egerton University. And so this is actually the 30 more, almost 30 years collaboration between Nanjing Agricultural University and Egerton University. So we start from 1994, supported by the Higher Education Cooperation Program, supported by the Ministry of
Professor.
Ministry of Education
Professor Wang.
of China.
Hello.
So I think everybody know,
Prof.
maybe you know this photo.
Hello, Professor
so
Wang.
This is Professor Wu Qingsheng. Many professors know him very well. We are Professor Wu from the same research group of mine. So he's actually my teacher. When I was a graduate student, he was the faculty member of our lab, and he stayed in Egerton University for almost 8 years. So we have established initiated many different programs starting from 1994. I think we have some landmarks for our collaboration, such as in 2012, we have the Confucius Institute, Professor Liu, Director Liu Yitao is now the director of the Confucius Institute, and this is also the only, the first Confucius Institute characterized, featured by agriculture. So we constructed, established such an institute, and it's very successful. And in 2014, so we are approved for the China-Kenya Joint Laboratory of Crop Molecular Biology. So this, we signed the MOU, and also this lab this lab was established. So the former Premier Li Keqiang also participated this ceremony for the establishment of this lab. Actually, this lab is not started from 2014.
Recording stopped. Recording in progress.
Hello, Prof.
The lab.
Your slides are moving.
And in 2019, so we, Chinese government, the Ministry of Science and Technology of China, approved the first 14 Belt and Road Joint Labs. So our lab, the China-Kenya Belt and Road Joint Lab on crop molecular biology, is among the first 14 laboratories. So this is a very successful laboratory, and now is I think I will talk about what we have done after the establishment of this joint lab. And after the joint lab, so we got a joint research project, also supported by the Ministry of Science and Technology. This is a national level, the National Key Research and Development Program of China. So we focus on our collaborative research on crops, on field crops, and also horticultural crops. And also in 2022, I think we have the Poverty Reduction Center in Kenya. And Professor, I think everybody know very well, but Professor also Li Yuan, is Professor Liu Gaoqiong. Yeah, we have generation to generation long history of cooperation. We are very happy to have this collaboration. So now we will introduce about some basic information about the joint lab. So our mission of this joint lab is fighting the challenges surrounding global food security, especially in Africa and also in Asia. So just now, the professor, the Professor Okori, he also mentioned about the challenges we are fighting, including the increasing population and also the climate change, threatening the crop production security. So, such as we have many abiotic, the crops is facing the abiotic stresses and also biotic stresses, diseases, pests, and drought and cold, and also flooding. So, and also we have limited natural resources. We have limited arable lands for crop production, and also the water and the fertilizer. And is also limited. So they are all limited natural resources. And except this, we have, for the crop production safety, we need to produce enough food to feed the increasing population of the world, global population of the people. And also we need to produce healthy people for such as we need to fight the hiding hunger, such the macronutrient deficiency. So many people in the world, especially in Asia and also in Africa, has such hiding hunger issues. So I just give some example for the crop production insecurity, such as the wheat stem rust, the UG99, the stem rust is a fungal disease threatening the wheat production. And also just one professor mentioned about, I'm very happy to see the maize leaf necrosis disease is also very severe disease in Africa. And I can see that one of the genome editing cultivars already got approved for release for production in Kenya. This is very good for fighting this disease. And except this, also such as the cold, freezing, frozen, and also drought is also very important abiotic stresses threatening the crop production. So for the construction of the joint lab, actually we analyze the mutual advantages and the disadvantages. So we need to know what we are fighting and what we have for the both countries in agriculture industry. So we have genetic resources, and also we have our featured production systems, and we also have well-established cropping systems, and also some research advances in research is supporting the crop breeding and crop breeding or crop production. And also we have the well-equipped facilities supporting the agriculture education, both from Africa and also from China. But also we have many disadvantages, such as the climate change, and also our agriculture system is very vulnerable, because we have very narrow the genetic diversity for the modern crop varieties. So we need to promote the basic and applied research on different crops to find new genetic resources, to develop the new varieties, to innovate new technologies, to enhance our agricultural capacity, and also to establish a well-trained talent research teams on agriculture. So our final goal is we are trying to achieve or approaching a high quality, high efficiency, and green and safe crop production system in Africa and also in China. Before we have this joint lab, we renewed the MOU between the two universities, and also we have mutual communications. We visit each other to get better understanding of both sides. So, and after our discussion and communication, we reached a consensus to construct the joint lab on crop biology. So this joint lab will focus on the graduate student and the young scientist training. The talent training is very important. So, and also we will apply and conduct the joint research projects, and also we will hold a joint conference, and we will have the technology demonstration, transfer, and commercialization. And also, we also would like to communicate or exchange the cultural agriculture is also, because it's different. We have different culture. Is also very important for the better understanding of the two universities or the two countries. So now the organization of the joint lab. So we construct the joint lab from both in Nanjing Agricultural University and also Egerton University. We have the academic committee and also administrative committee. So the committee members are from both universities. And also for the academic committee, not only from the universities, but also all two universities, but also we invite professors in China Agricultural University, Huazhong Agricultural University, the professors who are expertise in this area and also very expertise in international collaboration as our committee, academic committee members. So the director in the NAU is me. I'm working on with genetics and breeding, and director in EU is Professor Richard Muva. His research area is crop biotechnology. And also we have our administrative office, we have the PI teams, and also we have the core facilities to supporting the research, education, training, and international exchange and technology transfer, not only in the two universities, not only in Kenya, but also I think we can serve the research activities in other African countries or other universities in China. So just last June, just June in 2003, so Professor Richard Muva and other 6 professors visit Nanjing Agricultural University. We held a meeting, a joint meeting, and we have the committee. We appointed all the committee members for the academician committee and the administrative committee. So Professor Shen Qirong, he is also academician of Chinese Academy of Engineering, is the director of the academician committee. And Professor Chen Fadi, he is the president of Nanjing Agricultural University. He is director of the administrative committee. And Professor Muva, Professor Ogmino, and other scientists has also joined the academician and administrative committee. This is about the information, the basic information of the joint lab, we have the joint teams to focus on the research, joint research activities in field crops and horticultural crops. So we propose the 4 research activities in the joint lab. The first is genetic resource collection, performance evaluation, the trait evaluation, and also creation of the germplasms. And the second, gene mining, and elucidate the regulation mechanism for agriculture important traits. And also, the third is we innovate the new breeding technologies, and also we develop new varieties or cultivars for different crops. And the last one is we innovate modern crop production technologies, and also we demonstrate or transfer or commercialize these technologies. So we have the collaborative aspects in not only scientific research, but also tailored training, technique training, demonstration, and cultural exchange. So our final goal for collaborative goal is we'll find new genes important to for the issues we are facing for different crops, and we develop the new cultivars, we develop new technologies, and also we need to explore a new collaboration mechanism for the 2 sides in agriculture area. So now I will introduce about our achievements and progresses we have achieved in the following 6 aspects. The first is infrastructure development of the joint lab in EU. I think many professors already use this, has used or do your research or teach the students, undergraduate, graduate or graduate students in the lab. So I think this is a very good lab, and also this year, last year, because we have a key project from the Ministry of Science and Technology, we have some grant. We asked the professors in Egerton University what is the most needed instruments in this lab. And after communication, we also shipped some of the instruments from NAU to EU. I hope these instruments are in use in the lab. And also, we except the labs, we also have the agro-based science park. This is also very important for the demonstration and technology training of the crop, new cultivars and crop production technologies. So in NAU, we also have developed the infrastructure of the development of our joint lab. So we got the grant from the university and also from the Ministry of Science and Technology. And we also have the secretary, we appoint a secretary for to serve the joint lab. We have the office and the conference room, and we also have a website for this joint lab. So you can visit this joint lab to see the updated information. We update the information frequently. And this is so we also got some other grants to support the mutual exchanges and collaborative researches. So the most important joint research grant is from the Ministry of Science and Technology. And I will talk about some of our research progresses supported by the National Key Research and Development Program of China. So our research project, the title is Gene Mining, Germplasm Innovation, New Variety and Advanced Technology Technique Demonstration of major or main crops, including field crop and horticulture crops. So we have, we proposed 4 activities for the germplasm, for the gene mining, and for breeding, and also for the crop production technologies. And also we have the different research activities was conducted by different by the 2 universities. So the
Recording
right part
stopped.
is mainly in Nanjing Agricultural University. The blue is
Recording
mainly
in progress.
Egerton University, and the green is the joint research activities. So for the first activity, we have collected both in China and in Kenya, collected over 1,000 genetic resources or germplasms, including rice, wheat, maize, rose, cucumber, cassava, and millet, and etc. So we also evaluated the germplasms for their phenotyping, for their different characters, like disease resistance and quality and abiotic stresses. We use the different germplasms or the genetic resources we identified. We use them to mapping or identify new genes. So we identify the 86 valuable genetic resources, and we use them for gene mining. And we also use these materials not only for basic research, we also use these materials for breeding new lines with improved traits, such as wheat, cassava, cucumber, and pear, and also peach. So for the second activity, we not only the 2-side collaboration, we expand our collaboration with CIMMYT Nairobi, such as in maize, mRNA or ear rot, this Fusarium disease resistance. We collaborate, we have a 3-side collaboration to evaluate the disease resistances for these diseases. And also we identify the genes for the disease resistance. Except this, we also elucidate the resistance mechanism using multiple omics strategies. And for wheat, we focus on powdery mildew resistance, rust resistance, and also Fusarium head blight resistance. We also identify the germplasms for the different diseases, and we map the genes. We identify the genes on different for different diseases. And for rice, we also focus on the blast resistance, yield, and quality related traits. And we also elucidated the network for the different traits, such as blast resistance. And for the vegetable and ornamental and other fruit crops, we focus on the cucumber. This is a very good example for the collaboration. So we have international students from Kenya, from Egerton University. They work together with Professor Jinfeng Chen's lab for cucumber keratin synthesis regulation. We developed new materials, and also we identified the key genes regulate keratin synthesis. And we also developed new breeding lines or varieties for cucumber with a higher carotene content. And also we also work on this on the tomato, on fruit ripening regulation pathway, and also identify the genes regulate the tomato fruiting, fruit ripening. And for ornamental plants, we focus on rose and also on chrysanthemum. We have the genomic research, and also we identified the genes on disease resistance on rose, and also we identified the genes on the drought tolerance of chrysanthemum. And for fruit trees, our research is focused on pear and peach. So we have identified the pear genes regulate pear flower bud development and pear pollen tube development, because for pear production, the pollinating, artificial pollinating is a very important issue for pear production. So we focus on the identification of these genes, elucidate the mechanisms, and then we develop technologies to overcome the pollinating of pear. And the third activity is we develop new breeding technologies. So we have a very good technology in distant hybridization and chromosome engineering in different crops, such as in wheat, in chrysanthemum, and also in cotton, and also in cucumber. So we use wild species to hybridize with the cultivated species, and we can create new genetic resources for breeding new varieties. This is a very important technology for broaden the genetic diversity in the current cultivated crop species. And also we have marker assisted selection, and also genetic transformation and genome editing technologies for different crops, such as in wheat, in maize, in rice, and also in tomato and chrysanthemum, and different crops. We have this established such technologies. Just now, I also listened to a lecture about the regulation and in Kenya about the GMO. So it's will be very helpful to use these high throughput and high efficiency molecular breeding technologies for develop the new materials, genetic resources, and also breeding new varieties. And the last one is we innovate advanced crop production technologies. This is very in different crops, such as in wheat, we have the drip irrigation system, and for horticulture crops and also for rice, we have integrated management technology for water and fertilizer, such as especially in rice production. It's very high efficient. We can use a machine transplanting and integrated management of water and fertilizer. It will be labor saving and high efficiency, and also we can achieve higher yield. And also we have the dynamic growth diagnosis, monitoring, and regulation platform for field crops and horticulture crops. We have the smart agriculture research team for crop modeling system. And we also, for pear trees, just now I mentioned about this, for liquid pollination, it's very efficient pollinating technology. And for peach trees, we have a dominancy breaking technology. And it also can, not only can break the dominancy, and also can improve the yield of peach. So the outcome of collaboration project, we have published papers, especially we have 3 collaborative publications in cucumber and also in maize. Professor Chen Jingfeng's group and Professor Xiquan Gao's group with scientists from professors in Egerton University. We also have some patent and also so we also held, especially because we have after the joint lab, we have the pandemic of COVID-19. Even though we held many joint meetings for the two universities. So each year we have the annual meeting. So 2020, 2021, and 2022. So these joint meetings very helpful for our not only the scientists from the members from the joint lab, but also for the young scientists and for the graduate students. So we can understand each other better after this joint meeting. And we also have some joint meeting for the joint research project, and also we have some frequent communication between the two sides. So we also held many training programs or training on the smart agriculture, and also on the genomics, and also on the collaboration between our two sides, or between China and Africa. So this is I show some pictures in this June when Professor Mova and the other professor, Professor Liu, when they visit us, we have very many different activities. We visit different experimental stations to see the crop production, because rice production, maize production, ornamental production, fruit tree production, and also tomato, cucumber. So we have very a good communication, and I think this is a very fruitful visit. After the pandemic, we can see in person, we can talk face to face. It's a very good chance for communicate. And for talent training, we have the graduate students from Africa in the major in agriculture, and all in the agriculture college, and also in horticulture college. And we also have some joint supervising of some graduate students from Kenya, and also in Nanjing Agriculture University. I think this is a very good system for communication and also for tailored training. And also we have the Confucius Institute also help us a lot in the joint lab training and communication. And also we have some industries to participate, to join our collaboration. We held 8 training courses starting from 2019. So we have the beginning crop production technology training on rice production and the smarter agriculture. And we also have the joint lab workshop and technical training held on the webinar, not online training. So this is also very, very helpful especially for the graduate students. And this is some pictures to show the joint training courses. And this is for the training and also for the technology transfer of our rice technology. So we in collaborate with Wanbao industry. So we demonstrate, commercialize our rice production technology in Ethiopia. And now there is also a demonstration in Kenya. And also this is the dripping system demonstrated now in China. And this is a tea and soybean cropping system as demonstrated also in China. So all international collaboration draw close attention by the national and the international medias. So the CCTV and also different medias report, and also the People's Daily, Xinhua Night report our collaboration. So this is so the last I will talk about our future plan for the joint lab. So our joint lab will the approval assessment in the end of this year. So we will focus on the four parts. So first, we will strengthen our infrastructure building, not both in China, in AU, and also in EU. So we, especially now in AU, we still like sufficient space for international student training. But we will have a new campus in AU. We will move to the new campus. So I communicate with our president of our university. So we will apply more space for the international collaboration, especially for international talent training for students and also for young scientists. So that's for the talent training. I think we still have lack enough staff to support the routine lab running, because we have so many instruments. We need expert staffs who is very familiar with running of this equipment to support the education and research of the joint lab. And most very important is grants. So just the last talk is about the grants application. So still for our lab, we will our joint research program from the Chinese government this year will be end. So we need to apply new grants to support the joint research and also to support the routine lab running. So this is very important. So except from the government, the two government, and also from the university, so we need to attract more investment to our joint lab, such as industries, and also we need to support the research and education of the neighbor, not only in Kenya, but also neighbor African countries to promote a multi-site collaboration and to gain more international influences of the joint lab. So for joint training, so I would like to encourage the young scientists and also the undergraduate students or graduate students to apply the CSC international program. So this can, I think the Confucius Institute can help to pre-train the language training and also evaluate the language, and to access to help us to find the best candidates. And I think also the EU professors also can help us to recommend the candidate. And also I encourage the students to participate in our annual lab meeting and also training. To also encourage the young scientists and students to come to China for short-term visit, for short-term training. And not only in the lab, but you can also in the field technical training on smart agriculture, on modern crop production. And for the transfer and demonstration of new varieties and technologies, so we can communicate about the crop varieties on soybean or cucumber. But the most important, these varieties need to get the permission for commercial use in Kenya. And also for technologies such as the dripping irrigation system, and also the integrated management technologies on rice, wheat, cucumber. Cucumber grafting technology also is very helpful in fighting the disease and in cucumber production. So Professor Jin Feng Chen's lab is very expertise. They have very mature technology in this area. And also tea soybean cropping system, chrysanthemum production, liquid pollination of pear, domestic breaking technology, and peach. So after this, we need to promote multiple site collaboration in the area of agricultural research. We need to attract more participants from different institutes, universities, and also the NGO and also industries. So I'd like to thank the two universities for their support for the joint lab and our collaboration, and also the Ministry of Science and Technology, Ministry of Agriculture and Rural Affairs, Ministry of Education, and also Embassy of the People's Republic of China in the Republic of Kenya, Confucius Institute. Very important is thanks for all the team members from EU and NEU for their contribution to the collaboration and for the construction of the joint lab. So thank you very much. And this is show some pictures when I visit Kenya. They're very good memories, and I hope our friendship will long live China and Africa friendship. Thank you for your attention. Thank you.
Thank you. Thank you very much, Professor Wang, for your good presentation. Unfortunately, we were not able to see your slides because they were not changing, but we got you clearly well on how much you informed us about the various areas of collaboration between China, the whole world, and Kenya more specifically, and Egerton University in particular, for which, of course, we as Egerton University, we really appreciate. We've seen signs of that collaboration through training, capacity building, that is, and other things that you've done for us. I think I'll open the floor for some few comments and questions, if there is any, to Professor Wang, or comments. Any online? Okay, if there's no question, once more we want to thank you more sincerely, and let us give Professor one more round of clap.
Thank you very much. Thank you.
Thank you. Thank you very much. I think that marks the end of this session on joint research funding proposal development. And I want to leave the floor to the MC to guide us on the next area.
Thank you, thank you, thank you. Thank you very much, Dr. Steve. Let's appreciate him, please. Our participants who are online, please, we only have one presentation remaining, so hold on. So for this particular session, I would like to welcome Professor Boklan Bebe to chair the session. Welcome, Prof.
My name is Professor Bebe. I'm chairing this last session. We have been told only one presenter will be there. So let me confirm if Dr. Guchu is there. Dr. Guchu, are you with us? Hello. I also thought I saw her. Dr. Guchu, unmute. Even me. Ooh. Hello.
Hello.
Dr. Guchu,
Dr. Guchu, are you there?
Do you have Dr. Guchu online? Have we lost her? Dr. Guchu?
Dr. Guchu,
are you online?
Are you online?
Sorry, sure, I am online.
Aha, thank you, thank you. We want to have you come in now.
Okay, okay.
We want you to have you come in now, but allow me to introduce you to the audience. This is a session of capacity building, focusing on institutional managers and scientists. So we shall have Dr. Salome Guchu. Let me run you quickly about who she is. Dr. Salome Guchu is a proficient research and innovation policy management professional. She has orchestrated impactful initiatives at the Inter-University Council of East Africa. She has impressive career. Her impressive career includes founding the Kenyan National Innovation Agency and driving innovation governance projects. She has been appointed to United Nations 10-member group, where she is leveraging her chemistry PhD and diverse STI policy expertise to advance sustainable development. A mentor and advocate of STEM, Dr. Guchu champions inclusivity and progress in research and innovation. Dr. Guchu is going to talk to us on the topic of regional quality assurance and standards framework for research and training in East Africa. Dr. Guchu, may I welcome you to share with us your thoughts that should be able to build on the capacity we are aiming at. Welcome.
Thank you, Professor, for the introduction. Allow me to share my slides.
Please, yes.
Can you see them now?
We can see it. We can see it.
Okay, good.
Just put it on presentation mode.
Yes, it's now on presentation mode.
Not yet. Not yet.
Not
From our side.
Okay, let me first of all escape, then I...
Yes, that will work. Beautiful. Please go on. Oh.
It's now on presentation, right?
No, you changed it. Yes. Go on.
Okay, let me proceed. Okay, thank you for the invitation for this meeting. I'd like to say that in this presentation, I'll first of all would like to share about the IUCA itself, Inter-University Council for East Africa. Then I will give the perspective of quality assurance and standards, and of course the key principles, and I'll come towards the end, give some key considerations, and this will take into the case of the regional center that we are really glad that is being established at Egerton University, and congratulations for that. Inter-University Council for East Africa, I would like to indicate that this is an institution of East Africa community established under the EAC Treaty. And apart from that, it's an institution where universities within the region are members. And presently we have over 140 member universities. And glad to say Egerton University is one of the member institutions. And among the key things that IUC has been known of is on issues of harmonizing the EAC higher education systems. And this one is that one that had been acknowledged already by the heads of states, where they indicated that EAC should be, was declared it was declared harmonized for regional harmonization for education. And towards that, IUC has been developing a lot of policy documents, working with universities and stakeholders. We have regional quality assurance system, where most of institutions in quality assurance within institutions are also members. And it's also spearheading the East Africa Quality Assurance Network. And we have most of the academic members within the quality assurance department units within the institutions are members. We also forged developing of benchmarks, and we have a very robust quality assurance unit within IUCA, which work with all the universities, whether they are members or non-members. Participation is usually encouraged for all stakeholders. Other areas where we have also ventured into is areas of scholarships. And this one is important, especially to students, because we also partner with various stakeholders, provides graduate scholarship for students. And we also have a staff mobility program. And this can be obtained without going into details also from our website. We also coordinate matters of research and strengthening collaborations, and through various sorts of projects. And I would give some of the programs and projects that are coordinated within IUCA, like the Centers of Excellence projects, the program where even Egerton, you have a Center of Excellence where IUCA is a key coordinator in terms of monitoring and evaluation. So in brief, it was important to give that background. Now back to the main topic that I'm meant to present in this workshop. And I would like to share the perspectives of quality in research and training. And these perspectives are actually the perspective given by various stakeholders in academia, in research and training. And when we talk of quality, and it's something that we really need to think about, we think of excellence, which is mainly the view of academia. Right now, we will be establishing the center. The issue is excellence, and now we are aiming for excellence, and that's our view. But the key question is we have to be always asking ourselves, what is the view of the student and the employer? And that one should be a key focus. That is the issue of client satisfaction. And then apart from that, there is an issue of fit for purpose, because as we view the excellence issue, I'm sure by the time you are giving the programs that will be running at the RLC, we'll be talking of fit for purpose, because external assessors will come, will bring commissions for university education in case you consider various other accreditors. But that will be the external assessor will be looking at, is it, does it fit the purpose that is supposed to be, the goal that it's supposed to be achieving and the like. And then, of course, there is a endeavor to get accreditation. You want to be recognized even by industry partners, even internationally. So you have to look whether you meet the threshold. So that's also another eye that we'll be keenly focusing on. And then, of course, students, apart from whether they are satisfied, they will be looking issues of value addition. They are the clients. So there is added value. So these are the concepts of quality that we should be keen actually be looking at. And of course, from the morning presentations, and I had the kind of investment we are really looking into in establishing this center, there is a case where, of course, we'll all be sweating to talk of. And to prove, actually, this one is actually you have to be showing and sweat, and actually, if you are really, we will be really doing it. This is the view of the government, which is also part of the FADA. There is view of the taxpayer. There is view of the key funding, other key funding partners, including even the projects that will come up after that. So you have to be really looking all these aspects. They are all perspective of your quality in terms of your research and training. So I think with this slide, it's kind of give a ground perspective on various issues as we endeavor to establish that we really focus on. And the key question you'll be asking, why do we have to go down issues of quality assurance and standards? And of course, the overall is we must meet the establishment goals and aims in an efficient and effective way. This is general, yes, because mostly you are aiming to attract students, not just local students, international students. You want to interact scholars. So if there is no efficiency and everything is not handled effectively, if there is no quality, there are no standards in place, then it's not possible then to attract such kind of caliber of staff, student, partners, and the like. And now with that question, which we'll actually deep dive into it and answer more, there are key principles. And this maybe they may not be new, because being almost the last presenter of the day and having listened to various presenters, I've had the issues of governance and management. And this is not unique for the center, because you are also within the university. There is also there is a guiding framework that will be establishing this center. So the issue of good governance practices within that center, it's what will attract or repel partners, fathers, students, and any kind of stakeholder who will be interested to just approach the center. So this is very core and at the heart of every institution, and in terms of quality assurance and standards also. In terms of as a center, also among the key principles is institutionalize quality assurance structures, systems, and processes. That is in terms of support, monitoring and evaluation, and not just monitoring and evaluation. After that, there is a running and improvement. So I think this will be key. And this should be, it should be also will be, I believe this will be anchored also within also the main institution, and also to be in a position also to attract and engage with others. Of course, provision of conducive environment. This one, of course, comes in handy because the center is meant for enhanced learning, to be an enhanced learning environment, to be student-centered. So the approach must be, as we focus on how we are to implement the programs within center, it has to be very clear on how the kind of approach we want to look at. How do we incorporate feedback from students, from researchers, from stakeholders, provision of space, facilities, and resources. We know we talk of adequate, but adequate is relative. Nothing is enough, but of course, these are key areas that we have to really focus into, so that, and of course, engage all the stakeholders, incorporate all the relevant feedback. And because no one stays in an island, even at the moment, I'm sure most of the guiding implementation and how it will come up, I'm sure there are more or less in place on how we'll set up the center will be established, but you need to be also continuously open to listen to all other stakeholders in case something new that can be improved, even what already in place. Of course, I cannot forget to emphasize the issue of collaboration and teamwork, because this is where you get issues of continuous improvement and also benchmarking. And as I remember, this was highly emphasized also by the presenter, especially from the forum. And now we look at the case of the center itself. I'm now taking it's coming up to, and it's running. And here, the key considerations, issue of establishing a quality management system, which is very comprehensive. This is about institutionalizing, actually. The issues of document control, issues of continuous learning in terms of continual training and continuous professional development, because you cannot be focusing to attract if even the ones who are there are not also gaining on what is the emerging trends. And these emerging trends should not be just for the faculty, but it should include even those who are supporting in the laboratories, to encourage all areas to ensure that their safety procedures, all the consideration of quality are in place, and of course, people are working in a cohesive manner. Issue of equipment and facility management. And this one I would like maybe to indicate with, it's a key, it's an area that is quite pertinent, because sometimes we say we don't have a local, most of our local languages do not have a local word of repair and maintenance. And when we talk of equipment and facility management, think about how the maintenance of facilities, because that's where our institutions, our centers, most of the time we tend to go wrong. And I believe even in our homes, we always maintain. So let's put in place ways on how equipment and facilities would be managed, will be maintained, and the like, because there would be no need to have a laboratory with very very good equipment, a high caliber equipment, but once a small items just go down, it cannot be maintained, it cannot be repaired because of an item that is very small. Issues of quality control testing, that will go hand in hand with management and maintenance. Issues of risk assessment, compliance with ethical, regulatory, external and considering, especially when you start the programs. And we are talking of trying to attract international students and the like. Let's think of consideration of even obtaining, we'll obtain, of course, from our national regulatory bodies. But another thing which would be very critical is think of also in future how you'll get regional accreditation and or an international accreditation for maybe it may not be all programs, but maybe step by step, because that one is also a really good way of marketing, as well as see how you can get maybe for relevant industry bodies that also accredit, or even you can see they can be part of your marketing, a marketing way, because they start with you. And of course, collaboration and networking comes again now to specific institution. There is an issue of technology and digitalization. And this is very critical. This is a big center, which is not just a project, not just a program, but a lifelong initiative for the country. And it would be good to reinforce technology, enhance digital skill development within the relevant research and training. And of course, another thing is we must embrace flexibility in a constructive way. And the reason for this, when we talk of quality control measures, quality assurance and standards, sometimes it may require a lot of procedures. Sometimes even as we align also with the main institution, there are various procedures. But we should be flexible enough in a constructive way not to be overly rigid in some relevant areas, which will make you to stifle innovation and creativity. And in the end, you'll be limiting ability to adapt to new approaches and emerging trends that could enhance research and training outcomes within that center. So this one, of course, it's something that it have to be really balanced. And because we find that sometimes the rigidity goes hard with the creation of bureaucratic and administrative burden. So I think that this is that's the point, that you have to have control measures in place, but at the same time have the balance to avoid the creation of bureaucracy and administrative burden. And I believe as scientists and researchers, we understand when we see that the processes stifling the research, innovation, and creativity agenda within an institution. So these are things that has to be taken into consideration. And with that, being the last presenter, I'm very brief, and I would like to say congratulations, Egerton, and for the establishment of ICGEB RRC. And thank you for giving me this opportunity. Asante.
Thank you, Dr. Guchu. The club means we are very happy with the tips, so many tips you have given to us that will enable this center become excellent, achieve excellence levels. Thank you so much. Thank you so much for sharing with us. We'll have some few questions the audience may want to share with you. Do we have any of you with a question to Dr. Guchu? Yes, please go on.
Thank you, Dr. Guchu. This is Professor Molo from Egerton University. Thank you for the good presentation, the tips, as the chair said. I just wanted to get your thoughts. As we start, my mind has just gone beyond, because this will be a regional research center, and you have talked about ethical and regulatory compliance. To what extent should we worry about differences between different countries? And from your experience, are countries at the same level? And if they are not the same level, what could be the strategies of trying to, I don't know, I don't want to use the word harmonize, but maybe get around that to be able to have impact, particularly on programs that will be rolled out. Thank you.
Thanks, Professor Omoro. Probably the initial point that I would like to point out, of course, nowhere in the world that countries are the same level, and indeed, even within the region, within East Africa, countries are not the same level. But that doesn't mean that a program that cannot accommodate students from all from the region. And what I would like to really highlight the key consideration as you develop your programs, which like even from the university already, you attract students from the region, and endeavor to work with the institutions within the region, within Africa, and even beyond, because that's the only way that would be possible to also be known and participation also in various programs. But in terms of the issue of ethical and regulatory compliance, of course, the first thing is compliance within your country. And once you are already accredited within your country, within Kenya, already that's a recognized program within a country within East Africa. So, and these institutions, they also work together. The regulatory bodies within the region also work together. So as you endeavor, maybe if you want to go for an additional regional accreditation for wider audience, maybe sometimes they are bigger, you will find a bigger funding nowadays asking, do you have even an international accreditation? But it cannot be the first thing to begin with, because first of all, you have to get the national accreditations in order. Then as you grow, those international, regional will come in. But that does not hinder that if you are nationally accredited, you cannot attract. It will also depend with how you are disseminating and how you are carrying out your research and training within that center, and how you are attracting in terms of, like you usually say in Swahili, chema cha jiuza. As you start your program, let it be of high quality. Try to attract as many partners as you can, and also work with various institutions within the region. And there are various, within research, the relevant research institutions within the region, within the country. And also, this can be either initiatives of network at institutional level, and sometimes even at individual level. And that one, I don't think it's something that I would even lecture more, because that one happens even for as scientists, that you go create a network and bring it in, and eventually it becomes even an institutional partnership. So there is usually no hindrance in terms of, it's a matter of start at a very high level quality at a national level. And now, of course, looking at you are already international and regional, but endeavor to engage with regional bodies all the time, and also other institutions. Maybe I hope I've managed to answer your question very in the wrong close way.
Yes, you have very articulated your response very well, that we begin home, focusing ahead, and then we'll get it right.
Sure.
Thank you. Thank you. Any other more that we may pick? Is there any online? In the absence of other, would you take one from me?
Yeah, sure.
I like the idea that you bring on external accreditation for us as Egerton here, would using you, your organization, be external to us?
If I got you right, let me take you back to the recent IUCA actually initiated an accreditation, a regional accreditation program. It has started with a few programs, and there has been a call for institutions to submit. But you are aware also the accreditation, it also involves, it's a process, and it also involves resources. So it's something that once any program that an institution feels this one actually is ready to be accredited, it's something that you can it can also be submitted for accreditation at a regional level. And of course, we would also encourage participation of the researchers, the faculty within the institution, and even within centers to be participating also in to be regional accreditors. There are usually calls for regional accreditors because it's done the same way we do even at a national level from commissions, where we have the national evaluators who assess us for various programs and the like. So I think that there is opportunity for regional accreditation at East Africa level, and that one now is being done by IUCA.
Thank you very much. I want to ask them to give you another clap. We appreciate you as I hand back to the facilitating team.
Thank you, and bye-bye.
Okay, thank you, Prof. I think we have come to the end of our first day's workshop. And for those who are online, I request that we log in tomorrow, the same time at 9. The program is still on until around 4. We really appreciate your time, and I'm very sure that it has enriched you from wherever you are. Whatever that we have managed to discuss today, it has really managed to build you. Unless there's something from our team leader, maybe housekeeping or something. Okay. Then I really want to thank all the participants for the day. That is, those ones who have been here physically and those ones who have been there online. But please, let's be here tomorrow on time. Let's log in time so that we can also manage to finish in time. Thank you very much, and God bless you.
We want you to be there. Practical training that relates and fits so well in the current world.
Egerton, teamwork!
Egerton University, transforming lives through quality education.
I do remember Egerton University. Those memories fill me with gratitude. Egerton University placed me right inside a pool of professionals, gave me lawyers, doctors, business partners, everyone I need to continue advancing on speed dial.
I'm proud to be associated with this institution, Egerton, and it is shining, and anywhere I go, I normally meet people and they tell me they are from Egerton. Great people who have succeeded in different areas.
Success is like waking up to wash your face in the morning, only if you go through Egerton University.
We welcome those joining Egerton University. And for those not yet, what are you waiting for? Go to KUPP's website and choose Egerton University. There is a whole world waiting to hear from you. Let's give you a voice. Egerton University, transforming lives through quality education.
Egerton University houses the following